up. We are supposed to start 9 a.m., but as everyone, well, some of you may have known, there's been an accident just outside the complex, and that may have delayed some of our in-person attendees. Plus, we have quite a few people trying to sign in. So I'm going to wait about 5 courtesy minutes because I think I can factor it into my timeline, and we will start in earnest. I'm going to be talking for a while, so we'll start in earnest about 9:05. Thank you. Industry, as we call it, REdI, Annual Conference 2026. This year, our theme is that of innovative regulatory strategies to advance medical products. So, welcome to our in house attendees as well as our online attendees. Good day, good afternoon, or good night, depending on where you may be in your part of the globe. It's quite exciting to see so many of you here in person, and I know we will get even more people once they make it through the traffic and online here with us today. We certainly appreciate your commitment to attending this event from wherever you may be across the globe and overcoming any scheduling challenges. Please note the on-site Wi-Fi login information here on the slide. I see everyone with their laptops, so supposedly no one had problems getting in. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance Program, which operates within the division of CDER's, that's Center for Drug Evaluation and Research's, Division of Drug Information. It is great pleasure, along with the SBIA team and our colleagues, to be with you here today. I introduce Joe Tartle, Deputy Division Director for the Division of Industry and Consumer Education, DICE. That's Joe back there. From the Center of Devices and Radiological Health, CDRH, Joe will be our online moderator for the plenary session. And Dr. Lily Koo, Senior Biomedical Engineer of the CBER Advanced Technologies Program at the Office of the Center Director in the Center for Biologics Evaluations and Research, CBER. So in the interest of time, I'm going to skip this next slide, but I do want to focus on this geomap. This is a snapshot of the geomap for today's event. And this map really is quite exciting to everyone here at FDA, for it provides a very powerful visual as to the impact and extent of our outreach. As of this morning, we have over 8,300 registrants representing over 106 countries. Interestingly, over 47% of these registrants have self-identified as small businesses, and of those, over 33% report having 1 to 9 employees. Now, these stats continue to be extremely reassuring to us as they confirm that we are indeed reaching the small business sector of our audience, and thereby they also validate our mission and the very existence of the SBIA program itself. So let's now turn our attention to some logistics as it applies to everyone. In the part on your screen, in the part to the right of the slides of your screen, you will see links to the various session evaluations. These are only available during the actual session. So please take a couple minutes to complete those for us as the days go by. Our final evaluation for that day's event will be included in the thank you email that is sent the day following. So for instance, today, the thank you for today's event to all the people who attended online in person will be sent tomorrow. The certificate, when you complete the certificate, upon real-time attendance and completion of this survey, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. Now, the trick is that if you want that certificate, you must download it immediately upon taking the survey. Otherwise, the opportunity is lost. It does not allow you to come back in and take the survey again. Both the evaluation and the certificate are available only for 2 weeks. So for this REdI event, that means the deadline date is June the 3rd. As a reminder, all the recordings, the entire 2 days, are being recorded, all the sessions, and the YouTube links for both days will be posted on the event's webpage the day after the event concludes. Now, for those of you who have joined us via Adobe, You can submit your questions using the on-screen Q&A pod. For those of you who are joining us by YouTube, there is no mechanism to submit questions. Now let's do some logistics for the in-person attendees. Again, FDA Wi-Fi. We're going to have two mics, which I don't see just yet, but we're going to have two mics for the audience. And the way it works for the questions from the audience is that we'll go through and take two questions from the in-house folks, and then we'll go to an online question. And that, of course, depends upon the abundance of questions we have in-house. If we don't have enough in-house, we'll go ahead and take online questions. Um, Please note that our panelists can only address questions as they pertain to the content presented during these 2 days. Additionally, we will not be able to answer any application-specific questions. And as a reminder, please silence your phones, use the internet sparingly, as it may negatively affect our online delivery. At this point, I'd like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit as well. Special thanks to DDI's Division Director Mary Kremster, Deputy Director Catherine True, the REdI Planning Committee of Joseph Tartal, Tanya Wilborn, Michelle Sandrian, Kimberly Pumateo Kim, Lily Koo, Stacy Rivett, Lonnie Warren Henderson, all of our speakers and moderators or SMEs, everyone who assisted behind the scenes, writing scripts, doing everything. And of course, our DDIers. And most of all, you, because, if you did not have this need, we would not be here trying to bridge that gap and provide that need for you. Oh, I see them now. Sorry, I'm short-sighted.
There's one here and there's one on the
Yeah, okay, thank you, Joe. I was trying to see it in the middle. I came from the car this morning with my sunglasses on and wondering, why is everything looking so dark? If I had my sunglasses on, I definitely would see them at all. Okay, thank you. So let's see what's next here. Let's move on. Now that we've dealt with the logistics, let's segue into the plenary itself. Thank you again, everyone, for joining for what promises to be a very lively and forward-thinking discussion on how the FDA is evolving its regulatory approaches to keep pace with scientific innovation while maintaining our fundamental commitment to protecting public health. Most will agree that we are at a milestone in medical product regulation. The traditional paradigms that served us well for decades are being challenged, not because they're flawed, but because the landscape of medical product development is fundamentally transforming. Today's medical products are more complex, more personalized, and more data intensive than ever before. Our regulatory frameworks, therefore, must also evolve accordingly, and that evolution is already underway across the centers at FDA. This morning, we'll start by exploring how we are using innovative regulatory strategies and then discuss several important areas where these strategies are reshaping how we evaluate and monitor medical products. This includes using real-world evidence, the use of artificial intelligence in products, manufacturing and submission review, and alternatives to animal studies. Now, whilst these may seem like distinct topics, in fact, they share a common thread, each representing a shift in how we generate, analyze, and apply evidence to regulatory decision making. Let's further provide a little more context to these topics, shall we? Real-world evidence has moved from a supplementary data source to a cornerstone in the regulatory toolkit. We're no longer asking if we should use real-world evidence and real-world data, but how we can optimize its use across the product lifecycle, from supporting approval decisions to informing post-market safety surveillance. The challenge before us is ensuring that real-world data maintains the rigor and reliability necessary for regulatory decisions while still capturing the complexity of actual clinical practice. Artificial intelligence is perhaps the most disruptive force in technology today, and that includes regulatory science. AI tools are being embedded in medical devices, used to develop new therapeutics in advanced manufacturing, and increasingly being explored within our own review processes. Questions to yourself. How can we review as well as harness AI to make our reviews more efficient and consistent without compromising the expert judgment that remains essential to regulatory decision making? And how do we regulate AI enabled products when technology itself continues to evolve? Finally, alternatives to animal studies reflect both scientific advancement and ethical imperative. New approach methodologies from organ on chip systems to computational modeling are demonstrating that in many cases we can obtain better, more human relevant data without animal testing. The regulatory challenge is determining when these alternatives provide sufficient evidence to support product development decisions and how we can accelerate their validation and acceptance. What makes today's discussion particularly valuable is that we have our experts on hand representing each center. Dr. Michael Davis, Acting Director of the Center for Drug Evaluation and Research, or CDER, Dr. Owen Faris, Deputy Director of the Center for Devices and Radiological Health, CDRH, and Dr. Catherine Zarama, former Acting Director of the Center for Biologics Evaluations and Research, CBER. Whilst each center faces unique challenges based on the product it regulates, the innovative strategies we will be discussing today cut across all three centers. A breakthrough in AI assisted review in one center can inform approaches in another. Lessons learned from real world evidence in drug regulation can advance device oversight and vice versa. As we move into our roundtable discussion, I encourage everyone to think about these topics not in isolation, but as interconnected elements of a broader regulatory transformation. Ask yourself, how do these innovations complement each other? Where are they connected? What are the common challenges we face across all medical products? And where might center and product specific approaches be necessary? Our goal today is not to provide all the answers, that will be unrealistic, as regulatory innovation is a journey and knowledge fosters voluntary compliance. Rather, we aim to foster dialogue, share perspectives, and identify opportunities for collaboration as we work to build regulatory frameworks that are both scientifically rigorous, responsive to innovation, and ensure reasonably safe and effective medical products. With this context, I invite our experts to join me here on stage, and let's begin our discussion.
Testing, testing. I think mine's working, right?
Can you hear us okay?
Test.
Great.
Hello? Hello? Okay, so let's get to know our panelists a little bit better. Let's start with Mike. Mike, tell us a little bit, whatever you want to tell us about yourself, your role at CEDA. Tell us a little bit of, what interests you.
Yeah, so about me, I'm sure many people have read the news. So as of Friday, I stepped into the role of acting center director in CDER. So that's a big change. And I started. So I guess a little bit about myself. I'm a psychiatrist. I have had experience in the academic settings in VAs in clinical research, and trained in a clinical research fellowship. And I worked at the FDA in the Office of New Drugs Division of Psychiatry from 2016 to 2022. And then I left to work at a small nonprofit drug development company for about 2.5 years, and then came back as deputy center director in CDER. And as far as my interests, I'm very interested in advances in clinical trial methodology, and that was one reason why I took my former job in the nonprofit sector, because of some complexities involved in because of the methodological complexities involved in the types of studies that I was doing. And within CDER, I'm just really focused on strengthening the organization, supporting the staff, and just ensuring the high quality work of everything we do.
Thank you, Mike. Owen, let's go to you.
Sure. Good morning, everybody. Good to see everybody here. My name is Owen Farris, Deputy Center Director in CDRH. I've been at CDRH for almost 23 years, and I oversee a lot of the pre- and post-market decision-making for medical devices. And I just thought I'd spend a minute or so just introducing you to CDRH a little bit. So we work with a lot of small businesses. CDRH regulates over 250,000 different medical devices, and oversees over 25,000 registered manufacturing firms, the vast majority of whom are small businesses. On any given year, we receive about 20,000 medical device submissions. Most of those are under pretty strict timelines, so we are a very timeline-driven organization. And we really operate under two fundamental pillars, one of safety and one of innovation. On the safety side, a lot of that is about communication. And so just in last year, we issued 125 safety communications. And we also launched what we're calling the Early Alert Program, which is designed to notify external stakeholders earlier about what we think are likely future Class 1 recalls, getting what we know out to the public as soon as we possibly can. On the innovation side, we authorized 124 novel devices just last year. 44 of them were breakthrough devices, and 4 of them were from our Safer Technologies program. We also work with our colleagues in Medicare, CMS. And so this past year, we launched the Technology-Enabled Meaningful Patient Outcome Pilot, or TEMPO, which is designed to support the development and evaluation of digital health tools. We work closely with external stakeholders. So we work in particular with 17 different collaborative communities, most of which are comprised of patients, have patients and patient organizations in them. I know we're going to talk a little bit about innovation today. One of our focuses that we'll talk more about is what we call the Total Product Lifecycle Advisory Program, or TAP. And we now have over 100 innovators enrolled in our TAP program. We're going to touch on real-world evidence. We have over 250 devices that have been authorized in the past few years based primarily on real-world evidence. And just last year in December, we published an updated final guidance on real-world evidence and regulatory decision-making. In the regulatory science space, in the past year, we put out over 100 new regulatory science tools that help particularly new device developers figure out how they can evaluate their product and put that information into submissions for FDA. And we've also taken a lot of action where we find issues. And in particular, one of our focuses in recent years has been the focus on data integrity, particularly with certain test labs, where we've determined that the data can't be relied on. And we've been very public about that because this is a very serious issue. It's very important that the data that comes into the submissions to us, we need to be able to believe we can count on that. Looking ahead, we're really focused on our North Star of protecting patients, advancing technologies that serve patients. And really, that's really where we're focused all the time, is what is the next thing we can do to best serve patients. We want to continue to position ourselves to keep up with rapid technological advancements. There's a lot of that in the device space. I'm sure we'll touch on that some this morning. We want to continue to strengthen CDRH through evolving processes, leveraging of technology, and making sure that we prioritize our staff, making sure that we are giving them the tools and the support they need to do their jobs well and to serve the public. And we're going to continue to enhance our post-market safety efforts and optimize our compliance activities. And with that, I'll turn it back to you.
Thank you, Owen. And Catherine, what would you like us to know about your opening remarks?
Well, just that it's great to see everyone here today. Thank you so much for joining this time together at FDA. You know, it's a very unique time to be collaborating between CDER, CDRH, and CBER. And I'm so thankful I get to introduce you a little bit to CBER and a little bit about myself. So I got my start just like CBER at the National Institutes of Health. I was in the National Institute on Deafness and Other Communication Disorders. FDA CBER started in Building 29 on the Bethesda campus of NIH in 1988. We are somewhat of a startup, not yet 40, and still innovating with researchers along the way. When CBER moved to the White Oak campus around 2010, you know, part of our time was spent working with innovators, thinking about how they maximize their opportunities, and then really focusing their questions on regulatory science. I know you hear a lot about regulatory science. I hope you ask a lot of questions in each of the tracks that you get introduced to for what the impact on research might be for regulatory science. At CBER, regulatory science means that we're developing the standards needed to answer key questions about regulatory reviews. This includes our non-clinical work and even trying to introduce new approach methodologies. It includes our pharmacology, toxicology, clinical, and biostatistical reviewers. All of this together in a multidisciplinary review committee really expands on our opportunity to be creative, to seek collaboration, and then to find areas to innovate in. And it's really the way that we challenge one another, as Owen mentioned, in developing our staff, that I think we might be able to do a lot of really innovative product development pathways together with sponsors. And I hope some of you are here in the room thinking about those opportunities today.
Thank you, Catherine. It's always good to hear the pathways that, you know, everyone has chosen and to get here and to hear the passion that keeps you here. So let's stick with you, Catherine, and address the next question, first of all, to you. What are the centers doing to promote innovation, encourage collaboration, and improve patient access to medical products?
Well, I think on the innovation front, it's fair to try to introduce the product portfolio in CBER. Owen, you did a great job introducing that. Was very impressive for CDRH. For CBER, we carry about 439 products with supporting documentation, and this is everything from allergenics and bacteriophage. You might hear more from the CBER track if you join to learn about how we regulate bacteriophage development, the safety of the blood supply, cell and gene therapies, vaccines, and even xenotransplantation products. In 2025, CBER had advanced clinical trial efforts in xenotransplantation specifically. And then part of our innovation means, as I mentioned, that multidisciplinary team, but it really starts with the researcher-reviewer model. So I'm going to tell you a little bit about that. We have over 50 intramural research labs within CBER. Those researchers, again, start asking questions about what are all the potential opportunities that an area of development might take a product, either in non-clinical or in clinical stages of development, and then really seek out opportunities to de-risk that product development profile. And in collaboration with questions from sponsors or even really early developers through CBER's Advanced Technologies team, we start to dig into those questions side by side. And with the multidisciplinary review team, I think one of the things that we open up in collaboration is both formal and informal communication. So a lot of the tracks that you're going to see today and over the next 2 days will be a lot of those informal communications. And then the last thing that I would say about the innovation in CBER is really the opportunity for communications that speak to our accessibility, where we've been in terms of our guidance developments over the past year, how we've sought comments and opportunity for conversation in our advisory committee meetings. Our last advisory committee meeting was for the vaccine research branch for influenza in March of 2026. And then how we use that guidance and input from our committees to seek further developments in our policy process and ultimately our review decisions.
Thank you much, Catherine. Owen, let's go to you next. I know you alluded to many things in your opening remarks, so let's go to you next and see if you can expound on those.
Yeah, well, I was just thinking about Catherine's comment about the importance of de-risking. And when companies want to develop innovative products, having some predictability about what that process may look like on the regulatory side is so important. And so I'd like to just highlight a couple of programs in CDRH. The first is our breakthrough program, which has been around for a number of years, and literally hundreds of products have gotten authorization through that pathway, which is really focused on deeper engagement. Earlier engagement, rolling up our sleeves together and figuring out, how do we, for a novel product, what is the right level of testing? What are the kinds of testing that need to be developed? What is the regulatory pathway? And starting to map out some of that ahead of time so that there's more assurance that the company's moving in the right direction. We doubled down on that with our TAP program, which is even sort of a deeper dive to really be a very collaborative partner. And it is particularly useful for smaller, newer companies that may not even understand, how we're not the only player in the pathway to bringing a product to market. An important part is navigating FDA, but other important parts, and such as reimbursement and understanding the market and understanding how the product will actually be used when it is authorized are really important as well. And so in the TAP program, we've really been rolling up our sleeves together to figure that out with companies. And again, creating some spaces where we can, throw things on a chalkboard together and brainstorm so that we can figure out these answers together rather than have it be so formal. And we've seen that be really helpful in terms of bringing products to market. So maybe I'll pause there and give Mike a chance.
Okay, thank you much. Mike, how do you see it from CDER's perspective?
Yeah, no, thank you. Thank you. This is a really great question. So CDER is advancing innovation, improving patient access on several fronts, modernizing how clinical trials are conducted, embracing new technologies, and rethinking how we generate and use evidence across the entire drug development lifecycle. One of our most impactful areas of focus now is making clinical trials more accessible and efficient. And we're working to finalize guidance on integrating randomized clinical trials into routine clinical practice. And this is an approach that taps into the wealth of expertise that's already present in healthcare institutions and treatment networks. And this can reduce startup times and make it easier for patients with rare diseases or those that live far away from traditional trial sites to participate. And in 2024, we published guidance on decentralized clinical trials, which enable trial-related activities such as telehealth visits, electronic informed consent, and direct-to-patient delivery of investigational products to occur outside of traditional clinical sites. And really, this makes participation much more convenient and broadens the pool of patients who can engage in the clinical research. We're also advancing the use of digital health technologies, or DHTs, such as wearables, smartwatches, continuous glucose monitors, et cetera. And these allow researchers to collect frequent, objective clinical data directly from patients during their everyday lives. And this is particularly valuable for capturing clinical events such as seizures or arrhythmias that are difficult to measure in a traditional clinical trial setting. CDER is also actively fulfilling PDUFA 7 commitments in this space, including funding demonstration projects, hosting public workshops, and establishing a DHT framework and steering committee. We also have a public docket open through June 1, 2026, and we welcome in input from stakeholders on opportunities and challenges in this area, because this is a very high priority for us. CDER is also exploring drug repurposing as a pathway to address medical needs and improve patient access. And this is particularly for FDA-approved drugs where there's no current commercial interest in pursuing a new indication. And in this area, we have an open public docket on the topic that is open through June 11, 2026. And the information we receive will really help us prioritize disease areas and refine our approach to evaluating repurposing candidates and pursuing this priority. Beyond clinical trials, CDER is engaged in a broader transformation of regulatory science. We're shifting towards more human-centric approaches, such as recognition of new approach methodologies, such as organ-on-a-chip systems, computational modeling, and AI-driven tools as scientifically viable tools, alternatives to animal testing in certain contexts. And through the FRAME initiative, we're building a regulatory framework to support advanced manufacturing technologies, including continuous manufacturing and AI, with the goal of strengthening the supply chain resilience, and this also improves patient access to quality medicines. Finally, we're fostering innovation across the centers through the Rare Disease Innovation Hub, which was launched in the fall of 2024. And this brings together my colleagues here today, CDER, CDRH, and CBER. And through this, we address shared scientific and policy challenges in the rare disease drug development, which has many unique complexities. And we're also working internationally, collaborating with the EMA, for example, and we've developed shared principles for good AI practice in the drug development lifecycle. So altogether, I just want to convey that our CDER's efforts and commitment to a regulatory framework that's more agile, more collaborative, and more responsive to the needs of patients.
Thank you, Mike. So let's go to the next question. And this time I'm going to start with Owen. Sorry. Owen, how is FDA engaging in modernized regulatory science?
Yeah, thanks for that. So, I think, with medical devices, a lot of times there are, we do see some that are similar to previous iterations of devices, but we see a lot of devices that are really novel. And they bring new questions in terms of how can we evaluate those products, both from in terms of the clinical perspective, but also in terms of different bench tests that might be needed for those products. And so that's where regulatory science comes in. And one of the things we do, we have a whole portfolio on our website of what we call regulatory science tools. We literally have hundreds of these tools that focus on different elements of device testing where it's not a guidance, you don't have to follow it, but it's something that we've tested out and we think is generally useful for evaluating this kind of product in this sort of way. And then, many of those products become what I'll sort of describe as a little bit more certified in our medical device development tools, or MDDT program, where that's even a higher level of rigor where we've said this tool, you can use it in marketing applications and can be the basis of this area of testing and is likely to be very helpful for you. And we have many of those tools that we validate more each year. They can be really helpful in sort of providing some guidance because, again, a lot of the testing we look at is on the bench, it's in the laboratory, and it forms the basis of the many elements of a submission that comes in. I know we're also going to start touching on real world evidence, but that's another area where there's a lot of development I'm excited to talk about in that space.
You'll be getting there soon. Catherine.
Right. So I just want to accentuate some of the comments that Mike made and then also where Owen went. So from the CDER side, we're following along in CBER trying to bring about more modernization in our clinical trials. That includes the conduct and design that Mike mentioned. The e-consent, I think, is really exciting. And then also bringing in new statistical methodologies. So if you stay tuned for the CBER track today, you'll get a chance to see discussion of our draft guidance in Bayesian approaches led by John Scott in the Office of Biostatistics and Pharmacovigilance. You'll also get a chance to learn more about how we think about modernizing our facility inspections. And so if you stay tuned for day 2 on the CBER track, you'll get a chance to hear from the Office of Compliance and Biologics Quality, OCBQ, go through a couple of case studies. And then if you take a deeper dive on FDA's website, you'll get more information about FDA's PreCheck program, which also includes some modernization in facility inspections. And then I really enjoyed that you brought up the MDDT program. CBER and CDER have been collaborating on drug development tools, iStand, biomarker qualification programs, and really learning from one another, getting a chance to exchange opinions with the clinical and non-clinical review teams. So then the last thing I would say is probably if you get a chance to look at the playback from yesterday's rare disease endpoint advancement, the REdI program, sort of ties together what we're seeing in the non-clinical space and then trying to pull it forward into our clinical endpoints discussions.
Thank you, Catherine. And Mike.
Yeah, no, thank you. And yeah, so I covered some of these topics in my last comment, and my colleagues here also covered them. But but just want to reemphasize that CEDAR is advancing modernized regulatory science through a range of interconnected initiatives, and that really reflect our broader commitment to innovation, patient-centered approaches, and scientific agility. And I would say one of our the most significant shifts underway is related to the goal of reducing the need for animal testing. And in March 2026, FDA issued draft guidance formally recognizing new approach methodologies, like I mentioned before, is scientifically justified to animal testing. And this, I think, this marks a meaningful step in regulatory frameworks that center human biology at its core. Artificial intelligence is also being integrated across the entire drug development and manufacturing lifecycle within the industry, and CEDAR has developed draft guidance on the use of AI in drug development, and we're actively engaging with partners across the agency on digital health technologies and clinical investigations, such as my colleagues here today. And I mentioned our collaboration with the European Medicines Agency regarding the joint principles for good AI practice. And on the manufacturing side, I mentioned the FRAME initiative, which is preparing a regulatory framework to support advanced technologies in manufacturing, with a goal to really strengthen manufacturing reliability and supply chain resilience. And I mentioned breaking down. I think one theme I've really experienced across the last year is just enhanced collaboration across the centers, and I feel very blessed to be working with excellent colleagues in CBER and CDRH and on a number of things, and we're I think we're really functioning more as a cohesive team on a lot of these things. And the Rare Disease Innovation Hub is just one example of that. And in CEDAR's work, I would say that real world evidence and decentralized trials are becoming seen more commonly. And by expanding guidance on digital endpoints, real world evidence frameworks, and DCTs, we're really I think these initiatives can really broaden patient access to trials and improve the representativeness of patients in the clinical studies, and also generates evidence that more authentically reflects how patients live with and are treated for disease. So I think altogether, these efforts reflect FDA's commitment to build a regulatory science enterprise that's more agile, innovative, and patient-centered. And we're really trying to keep pace with the exciting and transformative advances that are reshaping drug development today that many of you are engaged in.
Thank you, Mike. And before we move on to the next question, I do want to make an observation to the audience. Pay heed to these resources that our panel are referring to time and time again, such as the regulatory science tools, open dockets, iStand. These are tools that are meant for your use. We encourage you to explore them. Don't just listen, but go and do some practical searching and explore them, familiarize yourself with them. This is one of the intent of these plenaries here. And also, I want to say that as you listen, you hear common themes: NAMS, FDA pre-check, rare diseases. And you'll see that those common themes have permeated through the agendas of the individual tracks as well. So look through those agendas and, kind of focus in on those particular topics that are of a special interest to you within your environment as well. So let's go on to the next question. And Mike, I will go with you first. How will FDA access relevance and reliability of real world evidence? That is the question.
Yeah, no, and this is a really good question because of the increasing interest in the use of real world evidence and in development programs. And FDA's approach to assessing the relevance and the reliability really begins with a foundational principle, and that is that the fitness of use is context dependent. And rather than applying a single universal standard, the determination of whether data are adequate must be evaluated in relation to the specific intended use of that data by the sponsor. And that said, for all study designs, sponsors must demonstrate that their data are sufficiently reliable and relevant to support a regulatory decision. And in that sense, this expectations functions as a universal baseline that applies regardless of the product type or the center that's involved. Within that universal framework, FDA guidance defines data reliability along three distinct dimensions. The first is accuracy, the closeness of agreement between a measured value and its true value. The second is completeness, the presence of all necessary data elements required for the analysis. This can also be an issue in more traditional means and missing data, missed patient visits and so forth, but also in real world evidence situations from clinical practice, for example. And the third is traceability, the availability of an audit trail or equivalent mechanism that establishes data provenance, meaning that the origin of a data element and the pathway by which it came to reside in an electronic health record or medical claim. A critical and openly acknowledged challenge is the absence of specific benchmarks or thresholds for what constitutes an acceptable level of data reliability for regulatory decision making. And to address this gap, FDA is actively funding demonstration projects to explore methods for measuring data reliability, including methods that could establish a minimum necessary level of reliability that is research question agnostic, meaning standards capable of providing meaningful guidance across a broad range of study types and regulatory contexts, rather than being tailored to specific use cases. Thank you.
Thank you, Mike. Let's go to Catherine.
Mike, that was an amazing overview. And maybe I'll just try and give a preview from day one on the CBER track. If you're following along, I highly recommend you check out our best innovative methods to put all of Mike's comments and components of evaluation in practice. So what was the goal of best innovative methods? Well, BEST stands for the Biologics Effectiveness Safety System. It's really our post-market surveillance for pharmacovigilance in CBER. And as I mentioned, there's a wide variety of products. So we're constantly innovating on how we can do better post-market surveillance. The IM, or Innovative Methods track, is really seeking to address the challenge of the EHR systems. So for those of you who get excited about medical billing and coding, I spent some time at the Centers for Medicare and Medicaid Services, but most people, when I start to talk about medical billing and coding, end up drifting off. So if you'll just bear with me for one or two more sentences, a lot of that structured data, really rigorously coded, often lacks the level of detail that we're ultimately seeking to make a decision about whether or not an adverse event really reflects the conditions that we need for effective post-market surveillance. And we know that there's additional unstructured information from physicians' notes or in the EHR, and really how do we unlock that in innovative ways? So using the principles that Mike already described and some innovative methods, we partnered in a collaboration with the Motiur Corporation to start looking deeper into developing algorithmically driven phenotypes and really challenging what we understand about the unstructured notes. Now, this is convenient because a lot of the review discussions that we have are analyzing hundreds of pages of notes. These algorithms do not fatigue. They do need additional input on what level of certainty might be needed, what level of completeness or quality is necessary to really determine whether or not we've met the conditions of a phenotypic algorithm. But if you go and listen to CBER's track discussion on Best IM, it's really trying to leverage large language models to better understand those algorithmically driven phenotypes to complement the coding information that we've already been able to develop in collaboration with our colleagues in CMS and international classifications of diseases. And so I think that's a great way to put into practice, all the principles that we share as 3 collaborative medical product centers.
Thank you, Catherine. I like how you tie everything back to the agenda. Owen, what can you say for this
Sure.
question?
Real world evidence is something that's really close to my heart. I could speak on it for a long time, but I will try to leverage previous 2 collaborators here on stage. One of my earlier roles at CDRH was the director of CDRH's Office of Clinical Evidence and Analysis, and I oversaw the development of CDRH's first real-world evidence guidance back in 2017. I'd like to just focus today on three recent activities and resources that are available for you all. So the first is that in December of last year, we updated that guidance. Everything that was in that before is still there for the most part, but we went deeper. And we described more examples, and we described in more detail what it means to be relevant and reliable in terms of your data source. And so I highly encourage you, if you're interested in a real-world data-based submission, to look at that guidance because I think it'll be really helpful to you. A second resource that we put out about a month ago or so is our second set of deep dive examples. A couple of years ago, we put out, I think, about 90 examples, and then last month, I think we put out another 70 or so. They're de-identified real uses of real-world data in device submissions. And they go through, what came in and what was challenging and how did we overcome those challenges and how did we think about those data in getting to a positive answer in the end. And some of those are expansions of label and indication. Some of those are using real world data for meeting post-market requirements. There's many different kinds of examples, different kinds of submissions, different kinds of devices to really try to hit at the constellation of the kinds of issues we see and hoping you can use that as a resource to help you come in with better submissions. And then the last piece I want to point to is something that is ongoing right now and you can help, which is we are collaborating in a public-private partnership with something called the National Evaluation System for Health Technology, or NEST, which is designed to support the development and use of real-world evidence in medical device submissions. And one of the things that they are working on right now is something called Nest Mark. And the idea is that if a data source, a real world data source, goes to Nest and they get Nest marked, then we will have greater confidence that that data source is full of reliable data. That it may or may not have the data that are needed for a particular submission, but the data in that data source should be counted upon as being true. And so right now, there is out for comment on the Nest website their algorithm for their proposed algorithm for assessing what should they be looking at to assess that a data source should get Nest marked. And so you can comment on that. We are hoping that toward the end of this year, we will roll out the first iteration of Nest Mark, and that in the future, as a device manufacturer, you can turn to Nest and you can say, I'm interested in doing a real world data submission. I want to make sure that I have a data source that is of high quality, that the FDA will have some reliance upon. And then if you come into FDA with a Nest marked data source, hopefully we can move quickly through the first steps of assessing, well, do we believe this data? And start getting to the questions around, is this data sufficiently complete? Does it have the right endpoints and information related to the question that you're asking? So that's the intent. I'm really excited about the possibilities of where we can go with NestMark going forward. And again, it's open for comment right now. Welcome your ideas.
Hint, hint, hint. Okay, thank you, Owen. So in the interest of time, I'm going to go a little bit out of sequence here. And I'm going to start with Owen on this question. How is AI impacting your work at FDA, very specifically with products?
Yeah. So, I do, as you noted, I think, AI impacts many different aspects of our work. One of the areas is actually the products we regulate. It also impacts other elements that I think we're going to get to with some of the other speakers. But we are seeing more and more products that come in with AI capabilities as part of what we review. And I think the important point is, I'll make a couple of points here. One is that we have to both think the same way we've been thinking and also think differently at the same time. And what I mean by that is that we are still looking at safety and effectiveness of products. We are assessing benefits and risks of products. We need to understand the patient population that product will be used in the context of use. We need to understand the alternatives that patient population may have, and as we're considering those benefits and risks. All of that is the same, whether it's a product that has AI or isn't, or doesn't. But there are some changes and differences, and when you think about an AI product that we have to dial in. And one of them is that unlike many of the products that we are used to reviewing, an AI product isn't static. It changes over time. It will ideally become a better product over time. And so we need to think about, how can we be sufficiently nimble so that patients have access to the best product they can as that product is getting better. One of the things that we have rolled out as a regulatory tool to help with that is something called the Predetermined Change Control Plan, or PCCP. And the idea of that is that when you come in with a submission for a product that may change over time, we can review not just the product that's in front of us, but your process for having that product change over time. And if we can come to agreement on that, we can come to agreement on what kind of changes might happen. How will you evaluate those changes? How will you determine that that improved product is in fact improved? If we can come to agreement on that plan, then we can authorize that with the authorization of the product, so that you don't have to come back to us every time the product gets just a little bit better and changes just a little bit, as long as we already agreed to the plan of how that would be changed and how it would be evaluated. And so that's a really exciting area. It's not just for digital health and AI-based tools, but it's an area that you can imagine it's very useful for. So maybe I'll stop there, but I think it's a really exciting area that we need to continue to be nimble and partner with external stakeholders as these products come in.
Yeah, it sounds like it's going to open up a whole new world. Yeah. Okay, Mike, can you address the same question, but from a manufacturing perspective, please?
Yeah, no, thank you. This is a great question, and yeah, so Owen, I think did a really good job of covering the use of AI from the FDA perspective in reviewing submissions. And one thing I wanted to highlight that really caught my eye recently is that an emerging as a new and emerging challenge that the FDA is confronting, and this is the inappropriate use of AI by regulatory industry and regulated industry. And just wanted to highlight a warning letter that was issued to a manufacturer in April that illustrates this concern in very concrete terms. Within the inspection of a facility, FDA investigators found that a certain firm had used AI agents to create drug product specifications, procedures, and master production and control records. And they did this without adequate human review of the AI generated outputs. And this is all presented publicly in a warning letter on the FDA website for reference. And the firm's quality unit failed to verify that the documents were accurate and actually compliant with CGMP in violation of 21 CFR 211.22. And perhaps most strikingly, when investigators informed the firm that it had not conducted process validation prior to distributing its drug products, which is a clear requirement, the firm responded that it was unaware of the requirement because the AI agent it relied on had never flagged it. And this case illustrates a fundamental principle that our FDA is now articulating explicitly within enforcement activities, that AI can be a legitimate aid in document creation and CGMP activities, but any output or recommendation from an AI agent must be reviewed and cleared by an authorized expert human representative of the firm's quality unit before it can be acted upon. And just as we discussed internally, over-reliance on AI, treating it as a substitute for human expert judgment, regulatory knowledge, or quality oversight is not acceptable and will be treated as a CGMP violation.
Thank you. We got some chuckles from the audience in that example there. Catherine, same question for you, please. Perspective of the submission review.
Right. So I agree with Mike completely. And I think CBER has been clear that these are tools really to aid the reviewers. And I don't have to explain to this audience that the complexity of reviews in CBER, because of the variety of products that we are carrying, often requires individualized attention. And I think it's important to note at this point that CBER is primarily focused, as you can see from data on our most recent approvals, more than 50% of the approved BLAs that are coming out of CBER are focused on populations of patients with rare disease. And so the combination of the complexity of the review and the individualized attention means that our reviewers, in order to aid in their review process, really can leverage AI as a tool in that development. And what do I mean by that? It's really formatting our checklists, extracting data and compiling it into a way that's interpretable and clinically meaningful, really cutting through to the heart of the review matters. And CBER has been looking at this specifically for review consults, whether that's through our intercenter consults with CDRH and CDER, or really just acknowledging that we have tools like model-informed drug development that require a chance to look here, look deeper. And AI is reminding everyone in CBER who's looking over these materials that in order to enhance your workload, in order to improve the quality of your review, really AI as a tool can remind us where the specialized areas of attention may lie.
Thank you very much, Catherine. So this session continues till 10:15 is what my agenda says. We would like to dedicate 10 minutes to audience questions. So let's wrap up the official panel discussion here. Let's open it up to audience questions. Please, anyone in the room? Don't be shy. Here we go, yes. Please address your question to the individual panelists,
Oh,
or
now
if it's a general question.
Okay. Yes, it's more of a CBER-CDER question. So there's been a lot of movement over the past year in integrating NAMs into review, and I think this is particularly important for small businesses because a lot of focus is at the non-clinical phase for these small businesses. And what I'm curious about is what is the opportunities you see to advance the non-clinical assessment. Thank you.
Of this discussion, as FDA was granted authority to focus on these non-clinical 2026, looking at novel approach methodologies for endotoxins testing. And you can see that guidance, how we're really trying to focus on alternative reagents that might produce better or more information for our non-clinical review, particularly in toxicology. I might also add that FDA had introduced guidance on next generation sequencing to look at off-targeting effects of genome editing. As you know, when you're starting to look beyond an individual animal to reduce the number of animals in testing, it's important to think about what question you're working to address. And the question that we're looking to address in an off-target manner really requires a integrated approach. And so looking at bioinformatics tools, looking at next generation sequencing techniques, really also emphasizing the utilization of CBER Hive, our super capacity for computing bioinformatics pipelines, have all given us more information to make these better decisions. And I hope that you'll get a chance to read some of the guidances that have come out in the last few weeks. Mike, is there anything you'd add on the roadmap?
Yeah, no, I think I think this is a really great question, and this is really a top priority for I think the entire agency right now, and we're looking at this through a number of approaches, and we are internally conducting regulatory science research to help build the evidence base to demonstrate when alternative methods would be good or better than animal studies, and we're also working on use of formal qualification pathways that can help provide the regulatory certainty for sponsors, which I think can be particularly important for a small business developing drugs. And internally, we're also very focused on training FDA reviewers within CDER to evaluate submissions that incorporate alternative methods and also to sort of support consistency across the review divisions in considering NAMs. And I mentioned before the draft guidance document on NAMs that came out recently this year, and we'll continue to focus on publishing new materials and reference information on the website to really help support the adoption of NAMs.
Thank you. Owen?
I would just say that in the device space, we are also really interested in figuring out, what tests can be done on the bench and in the lab without the use of animal studies, and in particular in the biocompatibility space. Like a lot of our testing that requires animals in the device space is related to biocompatibility. But we are exploring where, what other methods can be used to answer the questions, the key questions that are out there in terms of chemical characterization on the laboratory bench and that sort of thing. And again, I just point back to our medical device development tools as well, which are a library of testing methods that we have put out to help manufacturers figure out the optimal way to test the product for a marketing application.
Thank you, Owen, and thank you for that question. It gave us an opportunity to talk about a topic that we wanted desperately to get to. So thank you. Let's take an audience question here to my right, please.
Yes, hello. First of all, really great to have you all here together. I think that's a great opportunity, so thank you for this. My question has to do with AI and women's health. So we know women didn't come into clinical trials until the 90s, and we know that a lot of preclinical data didn't include female animals. So when we're talking about AI and generating information, how do we ensure? I'd like to hear the agency's thoughts on what are ways to ensure that this gap doesn't get perpetuated in terms of how AI is used now, and especially how AI is trained and how agentic AI is trained, given that there has been this gap. But at the same time, we know that evidence is being generated. We know that there is a drug that has sex-based difference in terms of labeling. We know that in terms of cardiovascular, there's data that specifically shows the disease manifests differently in women, especially with heart attack signs and things like this. So I'd like to get the agency's thoughts on how we ensure that the gap's not perpetuated with how AI is trained, and what kind of evidence you expect from sponsors in helping in that regard.
So I'll volunteer to go first, and then I'll let my colleagues chime in and clean up and improve. Thank you for adding the question. Also, thank you for recognizing, it's a lot to bring all three of the centers together. I think for the results of your first question, we're really thinking about how FDA and the US maintain its global leadership position, because these clinical trials are going to be global. The way that we approach our novel methods are going to have global implications. And FDA is really thinking about being a leader in developing AI frameworks. As I know Mike mentioned at the outset, the principles, the 10 guiding principles that are guiding our AI framework development have been done in collaboration with global regulators, including the EMA. I think keeping all of those principles applied to any one of our questions means that we're going to be making those considerations in ICH guidance development process, which then means that you're going to get multiple stages across multiple years with opportunities to collaborate and comment publicly on a number of platforms that are going to be addressing these challenges, including AI considerations. So I look forward to seeing what the future will bring, but I think the lens that we're trying to take is really thinking about FDA's leadership on an international stage.
Yeah, and so just to add, I think you bring up a really important point, because I'm familiar with the examples you brought up of the differences, the sex-based differences. I think you may be referring to Zolpidem, and there's other cases as well. And I think you raise a really important point that AI it really depends on the model that it's the training model, and if it is trained on a biased set of data, that can lead to then the output can be biased in the same way. And I was just thinking about your question about, so a new product if a new product is being developed, just as we recommend and essentially require sponsors to ensure the representativeness of the data to the U.S. population, I think that same I'm just trying to think of a scenario where just basically AI output based on a biased model would come into play. And I think it really depends on, I'd say, how that's being used. And I'd expect that in a sponsored development program that the actual data would include representativeness. the samples. And I think your point also goes back to the importance of having a human expert at the end of the line to review and carefully consider the output of the AI. And I think one consideration is how was that AI trained?
So in the device space, we see products really across our device spectrum we're seeing more and more AI. We see AI in mental health tools. We see AI supporting surgical robotic systems. We see AI in radiology and other diagnostic tools. It's really across the whole spectrum. And I think to your point, it's really important that the data that those tools are validated on be representative data. And that, again, as I spoke, some of these tools continue to learn even after they're authorized. We need to ensure that, again, that they are learning on representative data, and that their outputs are intended to treat that vast, spectrum of the population. So it's a really important issue. It's definitely on our minds, and it's a big part of how we conduct our reviews.
Thank you, panel. Joe, do we have any online questions? In the interest of time, in the interest of time.
That are using real-world evidence, are there any common gaps that FDA sees as it relates to problems with accuracy, reliability, traceability, or red flags that parallels when they provide their real-world?
Yeah, so I think, really all of those issues come into play. When we go through a review, we start with the reliability of the data. Can I believe the data that are in front of me? If I get to the point where, I have confidence that that data do represent what happened for those patients, I then have to start asking some of the deeper questions. Is there sufficient, are the endpoints that I'm interested in that are relevant to the question that's in front of me, are they in the data set? Is it sufficiently populated such that there isn't missingness? And if there is missingness, is it likely that that is missing in a biased way? Sometimes we're looking at outcomes that take months or even years to occur with patients. Well, does the data set have sufficient longitudinality, or does it drop out over time, such that, again, maybe the data that I'm looking at 1 or 2 years out is no longer representative of the original patient population, depending on how that dropout occurred. We also have questions around preservation of type 1 error. So in other words, did you pre-specify the question you were going to ask before you interrogated the data set? Or did you interrogate that data set many, many times until you got an answer that was interesting and you thought would support a marketing application? That makes a difference. And so we try to understand, the quality of your operation. Did you interrogate that data in a high quality manner like we would for a clinical trial? Now, I would say, sometimes people frame real world data as something lesser than clinical trial data because it's maybe not quite as structured and organized and prospective. And that it does have some limitations, but there are also ways in which it can be superior in other ways. We might have a much, much larger cohort of data. It might be more representative of patients that are actually in the real world that are going to receive this device or other technology. And so there are advantages to using real world evidence that we should leverage. But we do have to make sure that what we're looking at in front of us is of high quality, it can be relied upon, and it actually has the information that can help address the question.
Okay, thank you. In the interest of time, unfortunately, we'll only be able to take one more question from that first person there to my left. I'm so sorry to everyone else. Let's go with the one in-house question.
Thank you. Thank you so much, FDA, for hosting this very useful event today. My question is related to real-world evidence, so I want to stay on that topic, and really related to the NEST program, because this is the first time that I'm hearing about it, and I just also want to reiterate thanks for sharing all of the new useful programs that I will definitely look into after today. But in regard to the NEST program and with the revamped guidance, I am curious if the agency thinks that potentially there could be reduction in the need for pre-submissions with a 70-day timeline if we're NEST marked and if we're following the real world evidence guidance? Or do you think that it's still prudent to reach out initially with the FDA, have the pre-submission meeting, align on endpoints, and then get the NEST mark?
So great question. I'll break it apart a little bit. So my hope is that, if we're here a year from now, Nest Mark should go hopefully out toward the end of this year after we integrate comments. And that if we're here a year from now, there will be several data sources that are already Nest marked. A device company doesn't have to go find them. That there will be Nest marked data sources that will be on the Nest website and will be able to be accessible. And you'll know that if that data source has the data that are supportive for your application, that Nest mark will give some additional assurance that we are likely to expect it to be of high quality. And so that part of a pre-submission may be shorter or may not be needed. There may be other parts of your future submission that would be really helpful, particularly for a novel product, for you to come in and have a pre-submission with us. I think one of the pieces that's to me is so important is to make sure that we have the same understanding that you have about the strengths and challenges in your submission, what your device is intended to do, and so we can all hit the ground running and understand what it is we're looking at. And so I do think pre-submissions are extremely helpful. And when appropriate, I highly encourage companies to come in and submit one prior to a marketing application coming in. I also, to your point, they do take a couple of months to get through that process. And so I don't think a pre-submission is helpful for something that you already have the answer to and already have the predictability you need.
Thank you.
Thank you very much for your questions. And so let's wrap up officially. And I'll go through, each panelist and hear your final thoughts and your call to action to our audience. What message do you want them to take home and act upon? So let's start with you, Catherine, please.
All right, I know everybody picks come talk to us early, but maybe I'll say in the spirit of REdI, come meet the experts, right? So each track, you're gonna see 2 days from each center of the experts. These are individuals that are deeply engaged in the work on a day-to-day basis. They are thoughtful reviewers that have been contemplating these issues over many cycles of review. They are traveling to conferences to be able to engage in informal conversations, and they're excited to be here over the next 2 days because you've been able to come and reach out to them. So whether it's in your questions or your one-on-one connections, please continue to build those connections and come talk to people at the FDA that are the experts.
Thank you, Catherine. Owen, what would your thoughts be?
I'm going to leverage what Catherine said. And, I've been at FDA for about 23 years. I've literally been involved in thousands of submissions and across all of CDRH. And if I had to sum up the biggest challenge in one sort of statement that companies have, particularly smaller companies that may be coming to the agency for the first time, it would be summed up as tell your story. I think very, very frequently, and it comes up in so many different ways of challenges later, where a company, often a small team, has been deeply invested in a product and their validation strategy for months or years. And it's sometimes hard to remember and think about that when they're coming in with their engagement to FDA, that we have not, and that you are one of many other projects that we are involved in at any given time. And if you don't tell your story about what your product is, how you thought about validating it, why you think it's the right validation, the things you saw that didn't go well, and what you did to mitigate that, what data that surprised you in the first iteration of testing, and why you did this other test to show that the first test shouldn't be resulting in a concern. Telling that story, if in the absence of you telling that story, we make up our own story. We look at the data, we look at the device, we look at the description, we try to figure out how does this all come together into a story. And it's really helpful if we have your perspective. It's really helpful if as quickly as possible we can get to a shared understanding of what the device is, its strengths, its limitations, the data set, where there are holes and gaps, and how you thought about mitigating them. Let's get to the same shared set of facts, and then we can see if we agree. We can see if there are ways to navigate the challenges that are in front of us. So again, maybe I'll pause there, but I do think that overall, if you think, and as you're putting together a submission, have someone who is not deeply aware of your product and your submission read your submission. Do they understand it? Do they understand what you put together and why you think this is the right story? Now, very, very frequently, I feel like a submission comes in and says, FDA, here's what we have. Is it enough? And I really would caution and suggest that you come in with, FDA, here's what we have, and here's why it's enough. Here's why we think this is the right data set. Convince us, and then we can have a discussion about whether we agree.
Thank you, and Mike?
Yeah, no, thank you. I don't have a whole lot to add. I mean, I think a broad message that Catherine noted is and it sounds a bit trite, but talk to us early and often, especially I think when considering the use of innovative technologies, just to because I think that dialogue can really help optimize your chance for success. And I think and just to sort of summarize an overall message from CDER that I'd like to convey is that we're committed to meeting this moment with both ambition and discipline. And we see innovation as not optional because patients are counting on us to move faster, think more creatively, and build frameworks that reflect the science of today and not the assumptions of a decade ago. But rigor is also non-negotiable. And whether we evaluate an AI-generated manufacturing record, a real-world evidence study, or novel computational toxicology model, the standard remains the same. The evidence must be reliable, the methods must be transparent, and human expert judgment must remain at the center of every consequential decision. And so with that, I just want to thank the SBIA team for hosting this today. The SBIA is near and dear to my heart, having previously worked at a small company with 30 people. And so the SBIA was a regular source of information. So thank you so much for holding this event.
Thank you, Mike, for that endorsement. So let's thank our entire panel here. We appreciate your time, your advice, your knowledge. And let me just wrap up with some logistics and then we'll break. So I hope you guys take heed to what you've heard. And you got some wise gems today and take it to heart, especially these last, call to action items. So just some logistics. We're going to have break now until I believe, what time do we start, Corey? Somebody? 11. We're going to take that break because we have to pull the air walls and divide this entire room into 3 parts. Hopefully when you came in the registration, you would have been told to be seated into the room for which you have your predominant interest. So section A, room A would be CDER, which is drugs. Room B is devices, CDRH, and room C is CBER, which is biologics. So hopefully everybody is seated. You don't have to be disrupted and have to go find another seat or anything like that. The kiosk opposite the registration desk should be open for this break period, so you can go get some coffee and some drinks, and maybe they still have some donuts left over. So we'll see you back at 11. Thank you again very much.
Mic check. Mic check. Mic check. Mic check. Mic check.
So this is going to be your confidence monitor. There's what's in the floor. It's nice and white, isn't it? Yeah. Got some water here if you need it. And this is just your regular thing here. Forward, backward.
Okay.
And mic, just adjust it to you.
Okay.
And this is supposed to be the counter. I don't know if it's working or not.
It's okay. You can just let me know if I'm going too long or something. Signal me.
Let's put the pivot.
Awesome. And I'm told, just in case that's not working, I'm told 10:40, right? Is that, or sorry, 11:40. That way, I'll also.
Yes.
11:40 minutes of time for Q&A. Okay, perfect. Excellent.
So you started 11:10.
Yeah, so I saw.
Yeah,
Yeah. Okay.
yeah. I'm going to talk for 10 minutes again.
Okay. Have you not been out? Yeah. It's over. It's not.
Is it Raymond or Rahman? Rahman?
Yes.
Forgive me if I say it wrong in the heat of the moment. This.
Didn't make any sense.
Yeah. No, there's always something going on.
Right here.
But then you shifted over to.
Well, I was already here,
Oh,
which was like. I would just like more, like Oanh comes person. And I'm still, yeah, still feel lucky. Exactly. So are you a pharmacist by training?
Again, for pharmacists to do the shift for school.
Oh,
Did that.
cool.
Yeah.
Yeah,
So,
yeah, exactly.
Yeah. Yeah. Are you a pharmacist? as
No,
well?
I'm just like more like communications.
Communications. Yeah.
Okay, here we go. 1 p.m. No, not quite. 11 a.m. Don't know what time zone I'm in. Thank you again for coming in. And welcome to our REdI 2026 CDER track. Everyone here is in the right track, right? CDER track. And we're here at home on White Oak campus. So tell me, for whom is this your campus? Hi. About half and half, do you think? Yeah? Okay. How was the adventure with the traffic this morning and the accident outside? Yeah, I'm very happy that you guys made it through to the gate. All right. So some of the information I'm going to be covering for the next 10 minutes is going to be a bit repetitive if you were there at the plenary this morning. Speaking of plenary, did you get some good takeaways from the plenary? Yeah? Something that you're going to take back to your office and implement? Yeah? Can somebody, anybody, just one person, tell me something concrete that you're going to take back to your office and share with your colleagues or try to implement in an SOP or do something? Don't be shy. I saw a lot of heads nodding. True. Very true. All right, I'll take that one. Okay, so I know some of the information will be repetitive, but I also know some of our online people are probably just now signing on because of different time zones and things like that. So bear with me. Good day, good afternoon, or good night, depending where you may be, and welcome to everyone. Please note again, our FDA guests sign in. I am anticipating no one had problems in doing this. And I'll run through this. I am Brenda Stoddard still, haven't changed from plenary, with DDI and the Small Business and Industry Assistance Program. And that program has been around since 2010 within the division of DDI. My co-host over the next 2 days will be Dr. Corey Adair, and on AV is Will Tetlow and Gregory Cook, all members of the SBIA team. And now that a few more of us are logged on, hopefully online, let's do this poll. As we set up for the poll, I hope we're set up for the poll. Greg, are we set up for the poll? I try to do this every year, and I know many of you have been with SBIA quite some time, as I mentioned, going back to 2010 when we first started. And I hope that even so, many of you are also first-time attendees. So here's what we're going to do. A special welcome to our first-timers as we work through expanding our audience base and increasing our portfolio of SBIA events. And here's a bit of trivia. In 2010, when we first started, our very, very first REdI was here in the great room in section C. I recall it vividly. We had this one presenter that was due to speak, and 5 minutes before she was due to speak, she wasn't there. And I was getting very anxious. All she had to do was walk across from the building, you know, but I was getting very anxious. So I remember it very vividly. So that's it. And since then, we have not been back to the great room except for today, for this year, 2026. So here's the poll. The question is, five, C, 6 to 10, D, more than 10, E, I attended almost all since they started in 2010. So online, folks, evidently you have access to it online, so please go ahead and participate. In-house, folks. A, my first one. Hands up. Nice. B, 2 to 5. Okay, I don't have any prizes this year. I didn't have any. C, 6, have no budget, so no prizes. C, 6 to 10. We're getting smaller. D, more than 10. 2 hands. 2 hands. E, most since 2010. Charles, sorry, your hand should be up. Ah, there you are. Okay, you have a good contender there. I would have given you 2 prizes, but we don't have any. Okay, thank you very much for participating. Those online, thank you so much. We'll tally these up, and it sounds like fun, but it actually is a significant statistic in my report because we do take these attendance, to the conferences very seriously. It shows your interest, it shows your degree of participation and engagement on FDA's part as well. Okay, we're going to run through these logistics. We've done it before. I just want to highlight a couple of things. The session evaluations are only available during the actual session, the actual presentation. It's going to be on that pod next to the slides. Please take a minute or two to do it right then and there. Otherwise, you're not going to be able to do it. And we do want to get your feedback on the individual presenters and content. The thank you email will be sent. We're going to try to accumulate all the names of the people who attended in person as well as those online and send a thank you email the day after the day's event. So in that thank you email will be the general survey, not the individual session, the general survey. That's the one you need to complete if you want a certificate of attendance. And the certificate of attendance would allow you access, will support you for getting CE credits to RAPS, SOCRA, SQA, and ACRP. Again, download the certificate immediately upon survey because if you don't, there's no way for you to get it afterwards. You try to get in, it'll say, you've already taken the survey. So yeah, 2 weeks after, June the 3rd. As a reminder, all the sessions are being recorded. The YouTube links, so we have Adobe where you can come in through Adobe, and we also have YouTube where you can get the live stream. The YouTube links will be put on the website, the event's website, the day after the event concludes. Hopefully, because we have to put in a request and everything, that will be Thursday or Friday. Okay? For those of you who have joined us via YouTube, you can submit your questions using the on-screen Q&A pod. For those of you joining us YouTube, there is no mechanism to submit Q&As. For those of us here in person, this information here is self-explanatory. FDA guest, Wi-Fi, 2 mics. Depending upon the abundance of questions we have in-house, we would like to do 2 in-house questions, then rotate to 1 online question. If we don't have sufficient questions in-house, we go to online questions as long as we have those. And based upon previous experience, sometimes our audience will get very excited about a question or 2 in particular and may want to answer the question. Please allow the presenter to answer the question. And because that's why we're all here, to have FDA give you the FDA's perspective on these questions. As a reminder, silence your phone, use the internet sparingly. Yep. These are some QR codes here to access SBIA through our platforms. Please go ahead and scan them, and you'll be able to stay in touch with us. Upcoming events, June 11th. I'm happy to say that June 11th is the first of 3 installments that we're going to be doing for advancing generic drug development. This first installment is on bioequivalence challenges for patient-centric oral formulations. I want to go back to logistics for in-person. And let's talk about lunch, please. For those of you who pre-ordered through that pre-ordering system, we thank you for your order, and you should have received an updated email yesterday. That was Monday. Now, due to the increased number of expected on-site attendees, we had, to pivot a bit and change logistics so that everyone could not spend the entire lunch hour standing in line to pay. And so that particular pre-ordering menu is no longer valid. So for lunch each day, we will have be advertised on your Adobe through the breaks as well. So let's start our sessions, and I will introduce our first two speakers. First speaker is Meghana Chalasani, who is the Associate Director of Clinical Trial Innovation, and she will speak to us about clinical trial innovation from policy to practice. And then we have Motiur Rahman, Dr. Motiur Rahman from Office of Medical Policy, where he's a Senior Epidemiologist Policy Advisor. And he will speak to us about the framework to approval, operationalizing FDA's real-world evidence program in regulatory decision-making. So please help me welcome Meghana.
Thank you, Brenda. And let me just get settled up here. I'm sure I can advance the slides. Excellent. Good morning, everyone. Thank you all for joining us today, both here in person, it's a full house, and those joining us online. My name is Meghana Chalasani. As Brenda mentioned, I'm the Associate Director for Clinical Trial Innovation at CDER, and I lead our CDER Center for Clinical Trial Innovation, or C3TI. So for years, CDER has championed innovation, and our efforts to foster and support it span drug development programs, therapeutic areas, and disciplines. But the landscape that we're operating in has changed dramatically, and that change demands that we continue to evolve. We're seeing a greater focus on rare diseases and molecular disease subtypes. We're seeing a larger cross-section of the medical research community working together to advance clinical trial innovation. And there's a growing recognition that trials today need to leverage the most fit-for-purpose designs and tools to efficiently and effectively generate evidence that supports regulatory decision-making for safe and effective new medicines. In short, the case for innovation has never been stronger, but so has the urgency to scale it. With that in mind, in 2023 and 2024, CDER took a step back to assess the impact of the wide range of our various innovation efforts and programs. We conducted interviews, listening sessions, and hosted a public workshop with both internal and external folks, really trying to understand what was working across our programs and where the gaps were. What we heard was clear and consistent across three key areas. First, there was a perception of internal silos. External stakeholders wanted assurance that FDA pilot programs and review teams are aligned and sharing lessons learned. Second, there was a gap between policy or guidance and implementation. Stakeholders needed more concrete examples and resources to really bridge that distance between FDA guidance and real-world conduct of trials. And third, there was a desire for enhanced communication pathways, additional opportunities to engage with FDA on product-specific innovative approaches. And so CDER recognized these findings as an opportunity, not just to address the gaps, but to enhance the implementation of our innovative efforts and to maximize their impact on drug development. And so C3TI, or the CDER Center for Clinical Trial Innovation, was launched. C3TI's goals are directly responsive to what we heard. We aim to improve the efficiency of drug development through innovative clinical trial approaches by facilitating collaboration across CDER, enhancing engagement and knowledge sharing, and increasing structured engagement opportunities with drug developers. Everything that I'll walk you through today flows from these three goals. C3TI is a center-wide initiative, and we collaborate with and have support from a team of leaders and experts from across CDER. This includes the Office of the Center Director, the Office of Compliance, the Office of Medical Policy, the Office of New Drugs, the Oncology Center of Excellence, and the Office of Translational Sciences. And I'm grateful for their partnership and commitment to C3TI. Over the next few slides, I'll share how we're putting C3TI's goals into action. First, C3TI facilitates collaboration across CDER through its Innovation Forum, internal outreach, and a network of champions. Our Innovation Forum is central to this effort. It connects CDER innovation programs and experts quarterly to share updates, facilitate discussion, and encourage alignment. Forum participants lead programs and have expertise in areas like artificial intelligence, complex innovative designs, clinical trials with decentralized elements, digital health technologies, model-informed drug development, rare diseases, and real-world evidence. This collaborative approach ties directly back to the stakeholder feedback we received around coordination and knowledge sharing across the center. And to extend the reach of each of these conversations, we share updates and resources from the Innovation Forum through our internal newsletter, the C3TI Connection, which goes to all CDER staff. In addition to the C3TI Connection, we also leverage other internal outlets, such as staff meetings, to further promote awareness and engagement. Another key mechanism to facilitating the collaboration across CDER is our network of C3TI champions, who play a critical role. Our C3TI champions represent offices from across CDER, as you can see on this slide. They serve as liaisons for their respective offices, convene regularly to exchange insights and lessons learned, provide subject matter expertise to C3TI programs, and represent C3TI at external events. And on that note, I am going to put a plug in here for one of our C3TI champions. Dr. Cheryl Grandinetti, one of our C3TI champions, is going to be presenting later today, this afternoon, I believe around 2:10 Eastern Time. Her talk will explore how ICH E6R3's core principles, so this includes quality by design, risk proportionality, and fit-for-purpose clinical trial quality, how these can be applied in practice by identifying and managing critical to quality factors throughout the life cycle of a clinical trial. Enhancing engagement and knowledge sharing is another C3TI goal. And so to advance this goal, C3TI presents at events, hosts listening sessions, and convenes public workshops to improve transparency and strengthen engagement with external stakeholders. Since our launch, we've given over 65 external presentations, hosted 6 listening sessions with organizations representing various perspectives across the clinical trial ecosystem, and convened 2 public workshops. These external engagements have been invaluable for shaping our understanding and priorities. And so I want to highlight one engagement in particular, our most recent public workshop that we held a little over a month ago on scaling innovative clinical trial approaches, challenges, progress, and opportunities, and share some of the key themes that we heard that day. The first theme that came through clearly is that clinical trials are at an inflection point. The field is evolving, and stakeholders broadly agreed that progress depends on both execution and systemic change. Early cross-stakeholder collaboration and embedding research into routine clinical care emerged as priorities for improving accessibility and scalability. Building on that, there was strong consensus that the rationale for streamlined trial designs is growing. Experience from the pandemic demonstrated the value of simpler, more adaptive designs, and participants emphasized that rigorous upfront planning, regulatory alignment, and strong risk mitigation strategies are essential to maintaining data quality while reducing burden on patients. A third theme that surfaced repeatedly was the need for global alignment. Adopting innovative trial approaches at scale requires coordinating across regulatory authorities worldwide. And participants identified clearer, more harmonized international expectations and quality by design principles as key enablers. Finally, stakeholders emphasized that clear, consistent communication is critical to making meaningful progress. That means shared urgency, committed leadership, and well-defined problems across the ecosystem. Scalable pilot programs were highlighted as a promising mechanism for driving lasting, broader change. In addition to our external engagements, C3TI is also focused on building knowledge and fostering dialogue internally. Our C3TI Rounds is a speaker series that provides an internal forum for experts to discuss case studies and experiences with clinical trial innovation. We've held these sessions, about 5 of them, with external guest speakers and 4 sessions with FDA speakers, and covered topics like specific FDA reviews of innovative clinical development programs, registry-based randomized controlled trials, and quality by design and critical to quality assessments. We average about 650 attendees per session, which speaks to the strong internal interest we have in these topics. One thing that I personally especially appreciate about these sessions is how they reflect the multidisciplinary cross-cutting way that C3TI approaches all of our work, bringing reviewers, policy experts, and our inspectors from across the center together. For example, in our conversation with Dr. Adrian Hernandez from Duke University, so that's the screenshot shown here on this slide, on integrating trials into routine clinical care, we had experts from our CDER Office of Medical Policy who led the development of our guidance on this topic, participating alongside CDER clinical reviewers. More recently, we had a rounds on critical to quality assessments where we had our CDER inspection colleagues and a clinical reviewer joined by industry panelists actually who had direct implementation experience. To round out our engagement and knowledge sharing efforts, C3TI centralizes our clinical trial innovation resources, improving communication and access through its webpage, newsletter, and knowledge repository. We have both internal and external versions of our knowledge repository, which we call C3TI Compass. The external version, C3TI Compass, now has over 60 resources available across a range of innovative areas that you see listed here on this slide. So that includes resources related to AI, Bayesian approaches, trials with decentralized elements, complex innovative designs, digital health technologies, GCP, master protocols, integrating trials into routine clinical practice, RWE, and selective safety data collection, or SSDC. So C3TI's third goal, increasing engagement with drug developers, is addressed through our demonstration program, which expands structured engagement opportunities between CDER experts and drug developers. Our current project areas include Bayesian statistical analysis, selective safety data collection, and streamlined trials embedded in clinical practice, or STEP. The demonstration program emphasizes enhanced communication throughout development, with review teams part of the process from the very beginning. For example, when we receive a proposal for the demonstration program, the review team is the first to assess it and provide their recommendation on whether to accept or decline. This ensures that alignment between C3TI and the review divisions is there throughout the process. The program also recognizes the importance of timely engagement. We know that engaging and getting feedback requires time in a drug development program timeline, but we also know that speed is critical. So we've structured the program to be responsive. We accept project proposals on a rolling basis, provide preliminary comments on the innovative trial elements within our program acceptance correspondence, and design tailored engagement plans to support critical milestones such as protocol development and study startup. So it's an early engagement when it matters most. A guiding principle for the C3TI demonstration program is sharing lessons learned. We want to use the program to develop and disseminate case studies that support both drug developers and our review staff in applying innovative approaches. I'd now like to walk through each of our project areas, starting with Bayesian statistical analysis. So Bayesian methods offer a powerful complement to traditional statistical approaches in clinical trial design and conduct. They allow us to make probability statements about treatment effects, support ongoing trial monitoring, unify evidence across multiple endpoints, formally incorporate credible prior data into the primary analysis, and leverage all relevant trial data into subgroup analyses, making them a valuable tool for designing more efficient, informative trials. To be eligible for the C3TI Bayesian demonstration project, a trial must be a phase 3 efficacy or safety trial with a relatively simple design. So for example, a non-adaptive trial for one with a straightforward sequential design, such as a trial with the potential for stopping early for efficacy or futility. Within that framework, the Bayesian analysis can be used as the pre-specified primary analysis, as a supplemental analysis of the primary endpoint in the overall population or relevant subgroups or for trial monitoring. And so timely to this work, we recently published a draft guidance on the use of Bayesian methodology for clinical trials of drug and biologic products. This guidance covers a range of topics, including situations where Bayesian methods have been used, success criteria and operating characteristics, prior distributions, estimands and missing data, software and computation, and documentation and reporting. Our Bayesian statistical analysis project is led by experts from our Office of Biostatistics. We have 2 proposals that we've accepted and currently in this program. And then what you'll see is for each of our demonstration project areas, in addition to the trials that are in the program, there's other work underway to really support that broader adoption of these approaches. And so this includes guidance development, as I briefly mentioned, the relevant guidance here, case studies. In the case of the Bayesian project, we have examples of statistical analysis plans posted. We're always looking to hear directly from sponsors on their experiences implementing the relevant innovative approach. And of course, there's educational outreach as well. Our second project area is selective safety data collection, or SSDC. SSDC refers to a planned, intentional reduction in the collection of certain types of safety data in a clinical investigation. This approach is appropriate when a drug has a well-characterized safety profile and continued collection of common non-serious adverse events or routine lab assessments is unlikely to yield additional clinically meaningful information. In other words, it's about collecting the right data, not necessarily more data. To be eligible for the C3TI SSDC demonstration project, a trial must be a late-stage pre- or post-marketing trial for a drug with a well-understood and documented safety profile with respect to commonly occurring adverse events. This could include a trial for an approved drug seeking a new indication in a similar population, a trial seeking to expand the label to include additional endpoints in the same patient population, a safety trial investigating a very specific safety concern, such as a post-marketing requirement under FDAA, or a trial designed to provide additional evidence of efficacy where current data already supports a well-characterized safety profile. Supporting this project is FDA's final guidance on E19, a selective approach to safety data collection in specific late-stage pre-approval or post-approval clinical trials. This FDA guidance covers the general principles of SSDC, implementation considerations, practical guidance for sponsors, and its relationship to other relevant guidances and regulations. This guidance reflects FDA's implementation of the final E19 ICH guideline, which was published in September 2022. Our selective safety data collection project is led by experts in our Office of New Drugs. We don't currently have a trial in the program. However, we are working on broader initiatives to support the adoption of SSDC approaches. This includes publishing a white paper that included specific examples of SSDC and updating our review templates to promote awareness and help identify development programs where SSDC may be appropriate. We're developing case studies for our reviewers. We conducted a tabletop exercise with sponsors to hear their experiences and explore areas of uncertainty. This exercise was grounded in 4 specific hypothetical case studies of drug development programs. And then we're also promoting awareness and hearing feedback through our various engagement channels related to SSDC. Our third project is the Streamlined Trials Embedded in Clinical Practice, or STEP. So STEP encompasses trial designs that bring the trial to the patient. These trials may feature broad eligibility criteria that better reflect real-world patient populations, limited visits and procedures beyond what's already part of routine clinical care, decentralization of procedures that can be conducted outside of designated research sites, and use of real-world data to capture outcomes. Together, these elements can meaningfully reduce burden on study participants while improving trial efficiency. To be eligible for the STEP demonstration project, a trial must incorporate design elements that reflect clinical practice to improve efficiency, enhance patient centricity, while maintaining patient safety and data integrity. The trial must also fit one of 2 criteria. It's either in pre-market development with a reasonably well-defined safety profile, or it's a post-approval trial, whether sponsor-initiated or in response to a post-marketing requirement, where the population, procedures, and endpoints can all be appropriately incorporated into a large, simple trial. 2 guidances are particularly relevant to this project area. The first is FDA's guidance on conducting trials with decentralized elements, which provides strategies for bringing trials to patients. This includes the use of video and telemedicine visits, digital health technologies, direct distribution of investigational products, electronic informed consent, home visits, and engagement of local healthcare providers and facilities. The guidance also offers recommendations for designing and conducting trial-related activities at locations other than traditional clinical trial sites. The second relevant guidance is our draft guidance on integrating randomized controlled trials into routine clinical care, which speaks directly to the goals of STEP. This guidance envisions trials where design and activities are streamlined to align with clinical practice. Real-world data from electronic or other health records may be used to capture outcomes. Trial-related activities may be conducted as part of routine practice with participation of local healthcare providers, and dedicated trial staff may participate as needed for research-specific activities. Our STEP project is led by experts in our Office of the Center Director and Office of Medical Policy, including Motiur Rahman, who's going to be presenting right after me today during our session. We also have 2 internal work streams aligned with this project area. The first is the Integrated Compliance Workstream, and the second is the Modernizing Regulatory Requirements Workstream. And these workstreams explore how compliance approaches and the regulatory framework can be optimized to support clinical trial innovation, and are led by our experts in our Office of Scientific Investigations. While we did accept 1 trial into this program, and it had actually included an SSDC component as well, that proposal was later withdrawn. The sponsor had cited organizational changes, so we don't currently have a trial in the STEP program. As with our other project areas, however, we are working on activities to support broader implementation. STEP is another area where we had a tabletop exercise to better understand sponsor experiences around implementation. And then many of our C3TI rounds, this is that speaker series that I had mentioned earlier, have been focused on STEP-related topics. And then similar to the BAYS and SSDC projects, we're doing a lot of external education and outreach as well. If any of these project areas resonate with the work that you're involved in, I want to make sure that you know how to engage with the program. So a complete proposal includes 4 key components. First is general basic product and application information, including the product name, the pre-IND or IND application number, proposed indication, context of the development program, trial timeline, and whether the trial will be subject to review by other regulatory agencies around the world. Second, a list of discussion topics or questions you'd like to explore with CDER staff that's related to the applicable demonstration project area. Third, a high-level trial design overview, so a summary of the trial design, conduct, and analysis methods, with a focus on the data sources and proposed analyses most relevant to the project area that you're applying for. And fourth, an acknowledgement of our disclosure approach. As I mentioned, sharing lessons learned is a guiding principle of the demonstration program. And so if a proposal is accepted, details provided may be considered for disclosure, with the goal of sharing general principles and innovative aspects, while of course protecting proprietary or sensitive information. Sponsors can specify if there are any details that they would prefer that are not disclosed from their proposal. And so for full details on the submission process, I encourage you to visit fda.gov/C3TI, or you can also scan the QR code on this slide. So taken together, this slide captures the breadth of what C3TI has underway, spanning education, engagement, policy, and hands-on collaboration. And it's all grounded in a clear purpose: streamlining clinical trials, improving patient access, and supporting the adoption of innovative approaches that can get safe and effective treatments for patients faster. As we continue to build out these programs, C3TI is also taking a step back to ask, is it working? And so that's what I'd like to turn to next. So turning to how we're evaluating C3TI and refining our approach, we're continuously monitoring and tracking metrics across all of our activities to assess impact and identify areas for improvement. At the end of last year, we launched a focused evaluation to address a clear challenge. While industry expressed interest in C3TI's demonstration program, submissions to the program were lower than anticipated. We wanted to better understand the factors encouraging or discouraging sponsors from moving forward with proposals. Between January and February of this year, our evaluation team conducted interviews with experts, including regulatory policy leads, biostatisticians, and clinical innovation heads from 6 pharmaceutical organizations, ranging in size and scope to capture a broad range of perspectives. The evaluation confirmed that industry is genuinely interested in clinical trial innovation and values early input from FDA expert teams. However, we heard clearly that speed to market and regulatory predictability remain the primary priorities. Anything that appears to slow or complicate that critical path is viewed as a significant barrier. It was further revealed that while awareness of C3TI is high, the conflict arises in perception. We found that participation can be misinterpreted as a risky added step due to a few key areas of uncertainty and concern. Sponsors are wary of potential internal silos and uncertain about resourcing at the agency. We heard the need for assurance that our demonstration program and review divisions are aligned, clear differentiation between C3TI and other pilot programs and centers of excellence, and a better understanding of how standards and practices around clinical trial innovation oversight will scale across the agency. Next, sponsors find it challenging to translate high-level guidance into real-world trial implementation. The interviews made evident industry's appetite for more resources, concrete examples, and an interest in sharing lessons learned to bridge this gap. And there was a lack of clarity around the specific engagement opportunities and C3TI's role within the broader regulatory landscape by participating in C3TI's demonstration program. C3TI's intent has always been to focus on facilitating innovation by moving from policy concepts to real-world implementation. And so we look forward to incorporating these evaluation findings into our work and our program design. In addition to the demonstration program evaluation, we also conducted a training needs assessment, or TNA, to better understand what resources and support FDA reviewers and drug developers need to put clinical trial innovation into practice. The TNA was conducted in phases, starting with scoping and a review of existing resources, moving through data collection and analysis, and culminating in concrete findings and recommendations. Rather than duplicating what already exists, we were deliberate about building on the current landscape and identifying where the real gaps exist. The findings pointed to a few clear priorities. First, there's a need for more and better case studies. Industry told us they want concrete examples that bridge the gap between high-level guidance and real-world implementation. and our training needs assessment confirmed that externally available examples are limited. So we're prioritizing the development of new case studies, updating existing ones to improve usability, and exploring how to systematically identify case studies moving forward. Second, training resources need to meet learners where they are. That means organizing content, for example, by proficiency level, incorporating case study-based teaching methods, and creating structured discussion-based learning opportunities rather than one-size-fits-all for all resources. And third, access and dissemination matter as much as the content. We'll be leveraging existing channels to share the resources and then develop a strategy to ensure the content is relevant and accessible to the right audiences and build on feedback mechanisms to assess whether we're actually closing the gap. Taken together, these recommendations directly inform how we'll continue to evolve C3Ti's programs and support broader adoption of clinical trial innovation. So where do we go from here? We have a clear sense of our priorities for the road ahead. We'll be integrating insights from that recent public workshop I referenced that was held on April 14th to continue shaping and refining our programmatic direction. Building on the evaluation findings of the demonstration program, we'll be taking concrete steps to strengthen the program, addressing the barriers sponsors identified, and making it easier for drug developers to engage with us. We'll also be developing training programs informed by the training needs assessment recommendations, with a focus on giving both FDA reviewers and drug developers the practical tools and resources they need to implement innovative trial approaches. And we'll continue to build on and enhance our current portfolio activities, our cross-CDER collaboration, our rounds, our external engagements, while also expanding our internal and external outreach efforts to broaden awareness and drive adoption. Across all of these efforts, the through line remains the same: moving clinical trial innovation from concept to practice in a way that is efficient, accessible, and grounded in the needs of patients, sponsors, and reviewers alike. And before I close, I want to make sure everyone knows how to connect with us. C3TI serves as a single point of contact within CDER for non-product specific questions related to clinical trial innovation. Think of us as a switchboard. If you're not sure where to start or who to talk to, we can triage your inquiry and connect you to the right program or a right expert within the center. And so you don't need to have it all figured out before you reach out. We're a good starting point. You can visit our website, reach us by email, or subscribe to our newsletter using the QR codes on the slide. And perhaps we'll leave this slide up as I open the floor for any questions.
Thank you very much, Meghana. Yeah. Okay. Any questions in the room, please? We know how this goes. I have to pull teeth to get people to come up to the mics. Come up to the mics, please, so we can, we have people online as well that need to hear what your question is. Go ahead, please.
Thank you so much for this overview. I'm wondering if, my name is Izzy Stoll, Numera Therapeutics. I'm wondering, a lot of the examples that you gave, it really seemed focused on later stage clinical trials and post-authorization. And I'm wondering, with accelerated clinical trials design in urgent disease indications, rare diseases, and with orphan drug designation, how is it worth engaging on, say, more accelerated clinical trial pathways at the very early stage, say phase 1 or accelerated phase 1/2?
That's an excellent question. Sorry, I didn't catch your name. There was but excellent question. So I think there was two kind of components to that question. One, early engagement is always good. It's probably our most used talking point: frequent and early engagement. Come talk to us earlier and often, right? But I think that's a really interesting comment that you made, that across our demonstration project areas, it is on kind of that late stage pre-market and post-market. And that's correct. The innovative areas that we're looking to scale, these are, areas where we saw the most kind of bang for your buck, kind of where it makes most sense. We have some experience, but we want to gain more experience and so forth. We haven't really identified, certain therapeutic areas of interest or for example, saying only chronic or more common conditions over rare diseases or anything of that nature. But I think it is reasonable to say that most of it is kind of more focused towards the more common diseases. But once again, we're a good starting point if you are there. But I also want to put a plug in for our Rare Disease Innovation Hub at the agency, as well as our Advancing Rare Disease Cures, or the ARC program within CDER. I know Dr. Scott Winicky is going to be doing a session specifically on rare diseases and innovation in clinical trials for rare diseases, I believe tomorrow afternoon, if I'm remembering the agenda correctly. So I think he'll target a little bit more of the rare disease specific. So we are thinking about that a lot, but perhaps less within the current paradigm of the demonstration program within C3TI and more within the Rare Disease Innovation Hub and ARC program. But once again, this slide, if you're not sure where to start, we can still reach out to us even for some of that earlier stage engagement and so forth as well in the early trials.
Thank you so much. That makes a lot of sense. Thank you.
Thank you for that response. Anyone else in the room? Yes, please.
How about a common disease but a rare pharmacogenomic group?
I think this is probably the second most common response, which is it depends, right? I think we would need to learn a little bit more about what you have in mind and so forth, but perhaps I do think we have a lot of work there. And then we're also very well connected with our Quantitative Medicine Center of Excellence and the Model-Informed Drug Development and so forth as well, right? So we have a few different areas where that may be a good fit within the agency, but it kind of all depends on the context of use and a little bit more information about the drug development program.
Thank you. Do we have any online questions?
We do. So our first question is, recognizing the inherent challenges with rare disease drug development, what innovative clinical trial designs are being explored or utilized for rare diseases?
Excellent. I think that kind of pulls the thread on the first question we had from here in the room, right? So some of the approaches that we are doing, right, like things like Bayesian approaches, right, very much relevant to rare disease drug development, as well as our STEP, right? I know I've attended many patient-focused drug development meetings that are focused in the rare disease space, and we hear time and time again how difficult it is to, go to study sites, how burdensome it is both on the individual with the condition and their loved ones, and just despite all of that, we hear time and time again, and it's very inspirational that folks are committed to trial participation and contributing to research, but we understand that it's very burdensome. And so some of the elements that are incorporated as part of STEP, whether that is digital health technologies, decentralized features, really integrating it into routine clinical care so you're not having to travel, for example, all the way to an academic center of excellence and so forth. All of that is all geared towards trying to make trials more patient-focused, patient-centric, and improving the recruitment and retention in these trials. So there are certainly elements, I think, across all of these areas that are relevant to trials for rare diseases, but there's also a lot more as far as other innovative approaches that are particularly relevant to rare diseases. And so I'm going to wait for Scott to kind of speak to those again tomorrow afternoon.
Thank you, Meghana. I think do we have time for one more online?
Sure, we have time for one more online. Let me see here. So how is C3TI following the rapidly evolving landscape of decentralized trials and digital health technologies?
Great question. It is rapidly evolving, and we're trying to, kind of stay on top of it as well. We have our demonstration program, right? And so those two specific topics would fall under our STEP. And what we're hoping to do through the demonstration program is it gives us line of sight into how the implementation of these approaches are going. Because otherwise, we're not conducting the trials, right? But if we have an opportunity through our demonstration program to engage with sponsors who are doing this, we get to hear about their implementation experiences, what worked, what did not work, for example, as well as the outcomes and the output. But in addition to the demonstration program, this is an area where we have a lot of external engagement. For example, we have a public-private partnership with the Decentralized Trials Research Alliance, or DTRA, who've been thinking a lot about decentralized elements. And that kind of brings together drug developers as well as CROs and academics and a range of other stakeholders that are in this space. And so that's just one example. And then we've done exercises such as the tabletop with sponsors and other listening sessions to really understand and hear how the experiences are going, what's new, and once again, what's working and what's not, so that we can kind of take all of that back as we consider our guidance, as we consider the advice that we give to companies, and as we think about our education materials around this space as well. So we rely on you all basically to help us stay at the forefront in addition to what we're doing on our end.
Thank you very much, Meghana. So that wraps up this session. Thank you. And a round of applause. Excellent. Thank you. And now we welcome our next speaker, Dr. Motiur Rahman, to talk to us about the From Framework to Approval.
Thank you, Brenda. And thank you, Meghana, for introducing the topics. So Brenda introduced me. To introduce myself, again, a reminder has been quite I'm Motiur Rahman, Senior Epidemiologist and Policy Advisor in the RW Analytics team, sits in the Office of Medical Policy. It's a central hub or an incubator, if you'd like. And I lead the RWE review for CDER. And also I contribute to the international effort of harmonization, such as ICH, and supporting the training and process development. And I joined FDA about 5 years ago after in academia and industry. And I'm trained as a pharmacist with a PhD in pharmacoepi and a master's in statistics. I'll go next. So it's standard disclaimer, I don't have any conflicts of interest, and I'll mention some commercial products that has been approved based on real world data, real world evidence, but not necessarily an endorsement. This is just for educational purposes. So for my presentation today, I'll talk about a little bit how the RWD and RW came into being as a buzzword, and RW-related guidance documents with a focus on externally controlled trial, the common challenges that we face with the RWD submissions, and a few example cases of approvals and the lessons learned from there. So where did this RWD, RW buzzword come from? I mean, aren't we in the trial business, right? Even the previous presentation is a clinical trial innovation. So what changed lately? So this is what changed lately. So the interest in the real world data, real world evidence can be attributed to the improved access and the rapid analysis of the information, so-called big data. And the research showing that the observational studies can generate results similar to those of the RCT. And the popularity of the real world as a term, other factors came into being after the COVID-19, where we used a lot of real world evidence for our learning on the situation. And the 21st CARES Act of 2016 that mandated the FDA to evaluate the potential use of real world evidence for medical product approvals. But it's a point to be noted, often we get these questions that with or without invoking the terms real world data and real world evidence, the types of data sources and study designs are entirely new, and our evidentiary standards definitely didn't change. So for the 21st Century CARES Act, FDA established a program to evaluate the potential use of real-world evidence to support a new indication for a drug approved under Section 505C. So this is an important point. Support a new indication. So it was always meant for an already approved drug, we get a new indication or a label extension. But we have examples where new molecular entities got approved based on real-world data, real-world evidence. So it's a little bit of deviations from the originally planned, but a positive one. So I need you to focus on this. And also the satisfy the post-approval study requirements. Even before the 21st Century Cures Act, when the real-world data, real-world evidence was buzzwords, we have been using real-world evidence for the safety spaces from decades in the post-marketing setting. But now when the thing became more buzzword when we are using it for effectiveness. So in the pre-marketing approval, but the design, the fundamental epidemiologic design, statistical design, and analysis didn't change. Following that act, we drafted a framework describing the sources of real-world data, real-world evidence, the challenges, and the opportunities back in 2018. And we drafted and final guidances between 2021 and 2024. But again, the standards for the substantial evidence remains unchanged, or often we say substantial evidence of effectiveness, or in short, SEE. Still, we have the same standards. So FDA's RW framework for drugs and biologics of 2018, this is mainly applicable for the CDER, CBER, and the Oncology Center of Excellence, but the devices has a separate regulation and RW program, as you listened in the earlier in the morning, and they have a separate methods of to attack a problem. So that's usually our guidances are mainly applicable for these three centers. So established a multifaceted program for implementing real-world evidence broadly into the 4 categories: the internal agency processes, how we train within our folks, and how we implement this program as mentioned in the framework. This external stakeholder engagement, we go to the conference, and such as today, we are engaging with you guys so that we can share our thoughts on this area. The demonstration projects, it's an FDA-funded research projects. We have so far funded about 40 plus research projects, including 8 EO1. Those are the multi-million-dollar projects, so that we can learn as a shared learning from the academia industry, that where we are in the process, what methods need to be addressed, and what are the limitations. And of course, the most importantly, we work on the guidance development. So I'm not going into those definitions. You probably have read all those real-world data evidence definitions, where the EHR claims data, the disease registries are defined as real-world data, and the analysis of this real-world data generate real-world evidence, but often it is not limited to the secondary use of data. A lot of time, randomized control trials can also generate real-world evidence, as my colleague Meghana mentioned, the decentralized clinical trial or the point-of-care clinical trial, how the randomization happens into the doctor's office, and later on, the treatment received as part of the routine clinical care. So that's a little nuance when we think about real-world data, real-world evidence. Now I'm going to talk about our RW guidances, how we published so far, and their importance and contributions to the field. So far we have about 9 guidances. So the green marks that you see are the draft guidances, and we're still working on those. And the final guidance is being published for the EHR claims, registries, and the data standards. And also we have procedural guidances such as regulatory considerations and the submitting RWE to the documents. So a little bit on those. For the regulatory consideration guidance, we mentioned what to do and what to expect about when you submit a real-world data, real-world evidence, the data accesses. If the company does not own the data, if there's a third party involved who are going to submit the data, how to do that. So that's the focus of the regulatory consideration guidance. And the submitting document, it has a checklist, what needs to be in there. And in their cover letter, if there is a checklist of 12 points, that if those are included into the submission, it becomes easier for us to track and report on the real world evidence. The 3 guidances under finalization is the externally controlled trial guidance, which I'll go more in detail because this is the one single most used guidance, and the most of the submissions we see with real world data, real world evidence with the externally controlled trial guidance is very relevant. So I'll go over this a little bit more. Also, it's in the finalization process. We received a lot of comments from the public and we're trying to address it. Then the non-interventional study guidance is setting up the expectation, what we expect from a non-interventional study. And also the RCTs in clinical practice settings. So that we anticipate gonna be the second most used guidance in future, because that's, again, it preserves the randomization, the benefits of randomization, but also mimics the real world clinical practice. So these two highlighted, externally controlled trial and the clinical practice settings, or quote unquote, we don't like to use this term pragmatic trials because it is used a lot of time with a lot of meaning, but these two guidances are really important for our purposes. Okay, so for the considerations of the design and conduct of the externally controlled trial guidance, again, mostly used and most of the submissions we see related to this. So the focus of this guidance is the importance of the design considerations, such as finalizing the protocol before analyzing any data considerations for the external control arm, such as various comparability issues with the trial arm, and analysis considerations. We mentioned the standard practice, but FDA does not endorse any particular statistical method, but it's a general overview of the analysis consideration. And the fourth bucket, we say the considerations to support the regulatory review, such as access to patient-level data and engaging early. This guidance does not, however, address the other types of external controls, such as using summary-level estimates instead of patient-level data. This guidance does not discuss details about the design and analysis of the natural history study, nor the reliability or relevance of the various sources of real-world data that could be used as an externally controlled arm. And finally, this guidance does not also discuss the consideration of using external data to supplement a control arm of a traditional trial, as known as hybrid design. Many of those principles could be applicable, but still there are separate guidances for those purposes, especially the natural history or the rare diseases guidance. So what is an externally controlled trial? So an externally controlled trial is a trial that measures outcomes in participants receiving the treatment arm compared with an group that is external to the trial, as the name suggests. So it's not within a trial. That's what we understand generally, like a randomized controlled trial. So when should we use it in general? When we cannot do an RCT, or RCT is not feasible, or there are equipoise questions, or it is unethical to do. On those cases, we do externally controlled trials, and historical external controlled trials have been recognized as adequate and well-controlled studies, and we have examples of drug approvals based on these trials. Going more deep into the design considerations, for example. So in this guidance we discuss, I'll just go very briefly over this design. The sponsor should finalize the study protocol prior to initiating the externally controlled trial, including the selection of an external control arm and analytic approach, rather than selecting an external control arm after the completion of a single arm trial. And how many times we have seen they do a trial, then the trial fails, to meet their primary endpoint, then try to come in and leverage with an external control arm with a p-value less than 0.0005 and to say it a day. But it's not always the case. The trial arm, the external control arm should be selected way before probably the trial completion. We are seeing some improvements with the designs. We are disseminating this information, but this is an important point we need to remember. And the sponsor should pre-specify the plans regarding how to measure and analyze data on the important confounding factors and the sources of biases, and evaluating the extent of confounding and bias. And analytical methods to reduce such biases are very important for these type of trials, given it is a non-randomized design. And also need to understand the prescribing pattern in the external control population. For example, who in the eligible population are more likely to get the control drug? is there any particular pattern or not? And need to keep in mind with all those challenges and really get into the feasibility and to see that whether the external control study will be able to provide a convincing results when the effect size is large and the outcome interests are objective and well characterized. So in the examples, you'll see what do we mean by well characterized, objective endpoint, or it's a big effect size. Additional design considerations include the treatment attributes such as the potential imbalances in the external control arm derived from the RWD sources. Now, pause here. Often when we hear the word external control study, we always think, okay, this is an RWD thing. But often the external control arm could come from a prior clinical trials, right? The natural history could come from a prior clinical trial. So although it's not within the ambit of real-world data, the prior clinical trial has a more robust data capture. So that's often come into the play. However, unlike RCTs, important imbalances may occur between the trial arm involving the factors related to treatment, such as adherence, dose, timing of initiation, and the duration of treatment. And external control arm derived from the real-world data may lack detailed information on concomitant on the supportive therapies, as well as the characteristics and administration of such therapies, including drug formulation, dose, strength, routes, timing, and those are very not properly captured. Often there are misclassification within the real-world data. A very critical issue in the externally controlled trial is the selection of an index date, that is the time zero. If not designed or designated properly, can lead to the biased effect estimates and the immoral time biases. Any temporal differences between this date relative to the treatment initiation or other important landmark times between the treatment arm, especially when the external control arm is derived from the real-world data sources, can bias the treatment effects. And as for the immoral time, it is the determination of an index date in the treatment arm, and the external control arm should avoid analysis of a period of time known as the immoral time, during which the outcome of interest should not have been occurred. So failure to account this index date properly could bias the treatment effect estimates. In terms of outcome assessment, so the knowledge of a particular treatment by the patients, caregivers, or clinicians, or the investigator can potentially lead to the biased estimate. So whenever possible, we encourage that the outcomes should be assessed in a blinded manner. Often we get this question that how you're gonna do the blinder. So there could be independent blinded review. There's a possible operationalization. We can work together with the sponsor how to design a proper externally controlled study. Outcomes typically in the randomized control trials may be difficult to ascertain and evaluate in the real-world data sources, of course. In general, the outcomes are more likely to be recorded when the events are objective and require immediate medical attention, such as stroke or MI. Those outcomes are really well captured in the real-world data. But changes in the diagnostic criteria over time can introduce bias. So the non-contemporaneous control arm often using causes the problem. So we encourage to do a contemporaneous control arm if we have to do an external control studies. Again, the differential capture of the intercurrent events may make it challenging to interpret the treatment effect. For example, the initiation of a new therapy after the treatment drug of interest are protocol determined in the clinical trials, but in the real world data may not accurately capture additional therapies, and again, potentially confounding the treatment. Next is the data considerations. I'll very briefly say what's our thoughts on that. And so the guidance mentions that the data should be comparable across the treatment and the external control arm in terms of the time period, geographic region, diagnosis, prognosis, treatment-related factors, follow-up time, intercurrent events, outcomes, and missing data. However, the relevance of each consideration can vary on a case-by-case basis, depending on attributes of the treatment arm, the selection of the data source for the external control arm, and the stage of the trial, that is the design, conduct, and analysis. It's a standard go-to practice, but you don't have to check all those 10 domains for comparability across the trial and the control arm. For the analysis considerations, two very important things is the missing data and the misclassification of the data. These are very critical for the analysis consideration that we reported in this guidance. So the proposed analytical method should have a strategy for dealing with missing data, of course. Analytical methods such as strategies for imputing missing data may be used, but these methods require assumptions regarding the pattern of missing information. Assumptions about missing data can be unverifiable and may be difficult to justify, in addition to other assumptions required for estimation of the treatment effect, given it's a non-randomized design. An external control trial should be designed to capture and analyze for information relevant to the missing setting. External control trials should be designed to capture those, such as the available characteristics of patients with or without missing data. But missing due to the intercurrent events may interfere with the measurement of outcomes and estimation of the treatment effect. So the SAP should pre-specify how to handle those missing data. Another one is the mischaracterization can occur when the value of the measurement is assigned to an incorrect category, subsequently for analysis and potentially affecting the estimates of the observed drug outcome association. In real-world sources, different quantitative or qualitative description of the same measure can be assigned to different categories by different healthcare providers. For example, alcohol intake. If there is not a structured questionnaire, then it is very difficult how one physician going to capture it versus the other. Finally, the review considerations. The sponsors should consult with the relevant FDA review division early in the drug development program about whether it is a reasonable event to conduct an externally controlled trial instead of an RCT. For document access, sponsors must include in their marketing application the relevant patient-level data as required under FDA regulations, and for both the treatment arm and the external control arm. Again, if the sponsor do not own the data, a lot of time we see there should be an agreement with the data owners to ensure that the patient-level data can be provided to FDA. So as I described, this is how in general an externally controlled trial can be conceptualized. The bottom line is the data should be fit for use and a pre-specified protocol and SAP is required. Early engagement with FDA solves a lot of problem in the future. We have mechanism to engage early with the FDA, and we can suggest advised objectively where to go from there. Okay. So there are a few challenges, of course, with the real world data and the real world evidence. So the data source starts with the data source. The most important issue that we face in our day-to-day review is the data reliability and the relevance. So you heard Dr. Mike Davis was mentioning what's the reliability. So we say reliability is the accuracy, completeness, and traceability. So what do you mean by whatever we are measuring, are we measuring it correctly? In the real world data, often we say drug is in the market, then it goes to the physician's office, then it goes to the claims data or EHR. Maybe a drug formulation never had an injectable dose, but you go into the real world data, you'll say the same drug that only have in the market as the oral dose, it has been reported as injectables and other options, other route of administration, because these data were never meant for research purposes, right? These data were meant for bedside billing purposes. So that's an inherent limitations that we observe. So we need to have it is reliable. What it is measuring, it is actually measuring. The second issue is the traceability, where the data is generated from. So for our inspectors to go in and check whether it is captured properly, it needs to be traceable. So basically is the trace where it is generated from the bedside to when we get it as a CDISC or ADAM standard data. So that's like another topic of discussion for data standards. So that is important. Now, what is the relevance? So there could be some good data, maybe it's not relevant for this particular drug development program. For example, if you're trying to measure stroke as an outcome, and if the database missing information about atrial fibrillation, probably one single most important factors that causes a stroke. So that's a problem. Maybe that data captured everything correctly, but the most important covariate is missing in that data. And so that data is not relevant for these purposes. Missing or mistimed data. Now, missing data, I've talked a little bit about it, right? When the data is missing as part of their intercurrent event. So that issue is pretty common in there. So what do you mean by mistime? A lot of time in the real world data, we see a hit of a drug into the patient profile before even the patient is diagnosed with that particular disease. That's not possible, right? The patient needs to be diagnosed and then they will get the medication. So those are the issues that we see pretty frequently. Suitable capture of the endpoint data, there's the outcome data, and that's why we encourage to have a validation of what is being captured, what is not being captured. And the linkage of the data sources, a lot of time to answer a complete picture, we need to have a linkage about the EHR or the claims data and how this linkage has been done. In our EHR claims data, we have those descriptions. Okay. Then the design and the interpretation of the non-randomized study. I'm looking at my watch. So there's a residual confounding, problems with the index date, as I mentioned, and the inappropriate comparator. Now, the conduct of the non-randomized studies, that the protocol and analysis plan often is not pre-specified. They come after a failed trial, so that's an important. And a lot of time we hear there's lots of good data, but they do not have access to it, so it's not coming to FDA. FDA currently do not go into this data system and review it. So, so far, our reg says it has to come in-house, and we can work on that. Now I'll go over a few case examples where external control trials were successfully used. Okay, so these are the three key considerations from the real-world data framework, that the data should be fit for use, and the trial or study design used to generate real-world evidence can provide adequate and scientific evidence, and the conduct needs to meet the FDA standards. So there's the first example. It's an example of the program where we used a non-interventional study. So it was approved originally in the 1994 for prophylaxis of organ rejections in patients receiving liver transplant, then later approved for heart transplant based on randomized controlled trials. However, the drug was used widely, including the off-label for lung transplant patients. The sponsors submitted FDA for a lung transplant indication based on the findings from a non-interventional study, and the drug was approved. So the overview of the submissions, so the data source was from SRTR data on all lung transplant patients from 1999 to 2017, supplemented by the information from the Social Security Administration Death Master File. And this SRTR data captures every single transplant in the US. So that's an important point. So an observational study of 1 year to death and the graft failure from time to discharge was conducted. And the results showed improved in the outcomes of lung transplant patients receiving tacrolimus compared to well-documented natural history. So the key consideration, what was the substantial evidence of effectiveness? This quote-unquote RWE study was adequate and well-controlled. The randomized controlled trials of Prograf in the other transplant setting, including heart, kidney, liver, was the confirmatory evidence. And other randomized control trials showed independent contribution of tacrolimus in immunosuppressive agents. So the published RCTs where tacrolimus was used for prevention of organ rejection in lung transplantation are supportive but not required. FDA determined that the non-interventional study with historical controls to be adequate and well-controlled. Of note, the outcome of organ rejection and death are virtually certain without a therapy. And the dramatic effect of treatment helps to produce the preclude the bias as an explanation of the results. All right, going to the next example. So the first one was a CDER example. This is a CBER example. Is the approval of a gene therapy, Zolgensma, and it was evaluated for the treatment of pediatric patients less than 2 years of age with the SMA with biallelic mutation of the SMN1 gene. The sponsor submitted BLA to FDA with quote-unquote RWE study for efficacy endpoint of survival and seating without support, and was granted approval. For the overview of submission, the PNCR database and the NeuroNext databases, those are the two databases that was used for the data source. And the phase 3 single-arm clinical trial compared to the historical controls from those two databases were used. And the co-primary endpoints of the proportion of subjects achieving functional independent sitting for more than 30 seconds at 18 months of age, and the survival at 14 months were studied. And the results show significant improvement among patients compared with the natural history. Okay. So what were the, again, key considerations? What was the substantial evidence of effectiveness? The externally controlled trial, again, the RWE study was considered adequate and well-controlled. The efficacy data from phase 1 clinical trial were supportive evidence. Again, comment from the review is use of externally historical controls, although not ideal, appears reasonable in the case because an unmet medical need remained present. For the treatment of these fatal conditions, the natural history of infantile onset SMA is well documented. So that means it has a predictable disease course of action and follows a relatively predictable course. And the results of this study indicated that the expected treatment effect is large, readily ascertained, and shows close temporal association with the intervention. Now, I want to present one oncology example. As I mentioned, our designs are CDER, CBER, and Oncology Center of Excellence. It's the new indication of abatacept. It was first approved in 2005 and subsequently for the treatment of adult patients with moderate to severe active rheumatoid arthritis, for the treatment of patients 2 years and older, moderate to severe active polyarticular juvenile idiopathic arthritis, and the treatment of adult patients with active psoriatic arthritis. There was an unmet need in acute graft versus host disease prophylaxis, and the acute graft versus host disease, or GVHD, is a major cause of morbidity and transplant-related mortality following allogeneic hematopoietic stem cell transplantation, particularly in patients receiving the grafts from the mismatched donor. There are no approved drugs for this treatment, and the product received its approval in 2021. The database was used, the CIBMTR registry. It is deemed fit for purpose or fit for use because it includes data on every allogenic transplant performed in the US. This, again, everything goes into that database as mandated by law, ensuring completeness of the capture of the overall survival outcome. The randomized control portion of the pivotal trial demonstrated compelling trend but failed to meet its primary endpoint. A randomized comparison in the higher risk mismatched donor population was not feasible. The cohort was amended to a single arm study. Randomized to a placebo arm was no longer ethical, creating an evidence gap for this patient population. So an externally controlled study with overall survival as an endpoint on day 180 was conducted. The overall survival in the treatment group was 98% covered compared to the 75% in the control group. So again, what was the substantial evidence of effectiveness? The external control study was considered adequate and well-controlled, served as a compelling evidence, demonstrated large treatment effect, and leveraged fit for use high-quality registry data. The efficacy from the phase 3 study provided robust safety and efficacy data. Although the primary endpoint was not met, it demonstrated compelling positive trends. So in all three cases, you see it satisfies our 2018 framework that the study design, the real-world data source, and the regulatory considerations all met. In summary, two external control trials can be informative in certain situations, including rare diseases. However, methodological challenges exist. The FDA external control trial guidance provides a comprehensive overview, best practices. And the examples underscores the importance of early engagement, of course. It's an acknowledgement for the three centers who come together for the development of the guidances and the review. And a couple of important links I provided here for the Office of the Commissioner now periodically publishes or the RWE examples that's been approved. And if you have any question, contact to the CDER RW email box we respond, and if it is not relevant to us, we'll guide to the proper challenge. That I'll finish off my presentation. Thank you.
Thank you, Motiur. Okay, don't hesitate. Please don't leave the room. Come up to the mic. Any questions in-house, please? We have just about 5 minutes if we want to be on time. Go ahead, sir.
In the beginning, when you were talking about externally controlled trials, you made a comment about the treatment effect. I didn't quite hear it, but in all your examples, there was a large treatment effect. So generally speaking, those are easier to assess. If it's a minor treatment effect, it's not as easy to handle them, right?
That is true. Yes, and especially it is true because it's a non-randomized design, has some inherent biases. So the big treatment effects gives us a little bit more confidence.
That was short and sweet. Any more questions in-house, please? Remember, you're here for a purpose, to learn and to ask questions. Questions in-house? Are you guys hungry? Yeah? Okay, just a few more minutes. Do we have any online questions, please?
We do. So the first question for you is, can an externally controlled trial be conducted for a non-rare disease?
Yeah, that's a great question. So there's no easy answer to it, to be honest. But in the non-rare diseases, there's sufficient patient population to actually conduct a randomized study. And we have seen over and over again the advantages of randomization. It in itself takes care of a lot of biases. But when I say randomized study, means a well-designed randomized study. There are bad design randomized study as well, right? But randomization takes care of those confounders. We encourage to talk with the review division that whichever makes sense, but in the non-rare disease setting is still difficult. And we would ask, why do you think then randomization is not feasible or ethical, and why you want to conduct an external control study?
Thank you. In-house? Online?
All right, our next question states, what level of quality is expected from externally controlled trials where the control arm comes from a RWD source?
Yeah, again, as I mentioned in the presentation, that our evidentiary standard didn't change. Just because we are accepting or open to accepting real-world data does not mean it's an easy pass. The evidentiary standard is still the same, but a lot of time in the rare diseases, FDA exercise some flexibilities, but what level of flexibility, we don't have a specific tick mark. Okay, 5% flexibility, 20% flexibility. So it will be discussed with the sponsor and FDA that whether you're going to use the real world data as an external control or a natural history coming from a prior trial, whichever is the best suited for that particular drug development program.
I'm going to say this every time. In-house questions, please. Okay, do we have any more online?
Yes, we do.
Okay, thank you.
The next question is, what kinds of drugs or situations are best suited for real-world evidence submissions?
Wow, that's that's a loaded question. Again, I'm trying to avoid it depends, Meghana, so I'll not say it. But of course, the rare diseases, the examples in the FDA website, we have seen a lot of rare diseases. But for the external control, particularly if we understand the natural history properly. For example, we give this example that common medical knowledge is often can be used as an external control. For example, tumor doesn't shrink on its own or does not stop progression without an intervention. So we know the natural history that we need a treatment for that. So that's helpful to know whether natural history coming from a predictable course of action. So those gives us a little bit more confidence of using real-world data, real-world evidence.
Okay, my clock says 12:30. Thank you very much, Dr. Rahman. Before we do adjourn for lunch, remember we have the one-to-one sessions. Our speakers will be here. Motiur, I guess you could take this side. Meghana, you can take that side. If you have questions, it's very informal. just come and ask your question. We do have the caterers outside there set up. We do have the kiosks. Grab your lunch. If you have a question, come in and ask. Try to eat first or after, please. It makes asking the question easier. So, and same goes for the presenters if you want to grab some lunch before or during. And okay, so and we'll see you back at 1:30. It is, yeah. No, what time is lunch? Yes, 1:30. Thank you very much. Good afternoon, everyone. And I hope everyone had a really good lunch. But I'm wondering whether we should have had such a good lunch because now everyone wants a siesta. And we can't afford to do that. We still have many more, at least 4 more presentations this afternoon, and they're all equally important. So let's start. I know some people may still be outside enjoying the weather. And pretty hot, wasn't it, outside? Yeah, yeah. Tomorrow, I think it's going to be a little bit hotter. But let's start. Welcome back to everyone, those in-house and those online. It's my pleasure now to introduce the next 2 speakers, after which we will break from 2:50 to 3:05 p.m. Remember, the kiosk is not going to be open in the afternoons, so this will be a good time to network or walk around where you can and get some of that lunchtime, get some exercise after lunchtime. Okay, so our next 2 speakers before the break would be Dr. Anastasia M. Shields, actually Commander Shields of the U.S. Public Health Service, and she is with the Office of Human and Animal Drug Inspectorate 2, Branch 3 in the Office of Inspections and Investigations. And Dr. Shields will be speaking to us about quality assurance through CGMPs, understanding FDA drug manufacturing inspections. Commander Shields will be followed by Dr. Cheryl Grandinetti, and she will be speaking to us about ICH E6 Revision 3, Achieving Quality Through Purpose-Driven Clinical Trials. Dr. Grandinetti is the Associate Director for Clinical Policy within the Office of Scientific Investigations at the Office of Compliance within CDER. So help me welcome Commander Shields. Thank you.
All right. Good afternoon, everybody. I'm so lucky to be the speaker after lunch, so I'm going to do my best to keep you all awake, okay? I know how after lunch feels. So we're going to talk today about FDA inspections. I do a lot of FDA inspections as my everyday job. I've been doing them since 2014, and I've seen a lot of things and been to a lot of firms all over the world. We're going to cover the FDA's core regulatory expectations, and hopefully you'll better understand an investigator's inspectional approach after this presentation is over. We're going to cover how to evaluate organizational readiness in a review and what FDA observations on a 483 are made of and what their meaning and relevance is. And then we're going to look at best practices to strengthen your inspectional preparedness and your compliance culture at the firm and better understand how to respond to FDA 483 observations. Okay, so the core of all of this is 21 CFR parts 210, 211, and 212. Okay, this is the foundation of GMP expectations. So all manufacturing operations have to conform to 21 CFR. So what does this mean? It means your facility, your equipment, your personnel, processes have to meet CGMP standards. Compliance isn't a point of time, moment thing. It is an ongoing process. It never ends. It is grueling. And depending on the size and complexity of your facility, it can be very difficult. So the C in CGMP means current. So we expect facilities to maintain current GMP practices, not GMP practices that were relevant 20 years ago. If those have been updated with something that makes drug products more compliant, then we expect that your facility will have updated your practices to follow the C in CGMP. So the C in CGMP, we expect you to maintain a robust functioning quality management system. That quality management system has to drive decisions at every level of the organization. You need to have a quality unit. Every facility should have some form of a quality unit. Okay, and I say that because I've been into a lot of facilities where there is next to no quality unit and it's diffused amongst other units. And it makes it very hard for them to maintain a system of quality and organize a quality structure within the organization. The quality unit should have the authority and independence to approve or reject material, processes, finished products, and to make decisions that are not overridden by production or business pressure. One of the common deficiencies that we find are policies and procedures and practices that are applicable to the quality unit aren't reviewed or approved by the quality unit. So this is your SOPs, your validations, your investigations, all of these very important elements of a drug manufacturing facility and a drug manufacturing infrastructure will come across situations where, let's say, for example, I find an investigation and I start to ask questions, and someone from the quality unit's there and they have never ever heard of this investigation, and it's never come in front of their desk. These are situations that can lead to problems within the quality system. And the quality system governs most of the operations that occur at a drug manufacturing facility. So another thing is equipment qualifications, process validations. These need to stay updated. All right, this is a common deficiency that we find when we go into a firm for drug manufacturing inspections, is there may have been an equipment qualification done 25 years ago on an old piece of equipment, but since then, that piece of equipment has been changed or it's now being used for something completely different. So it needs to be qualified for its use. Another important thing that we still focus on for data integrity is ALCOA. All right, I'm sure a lot of you have seen this acronym. It still applies and it's very relevant. Validated manufacturing processes and equipment qualification. Your manufacturing processes at a drug manufacturing facility need to be validated to show that your product and your process can produce a finished drug product, an API or whatever, an intermediate, whatever you're manufacturing, that is consistent and that it adheres to internal specifications and already set specifications, okay? This is a big part of GMPs. The process validation studies that are done on products need to also look at intra-batch and inter-batch variability to determine if your process is maintaining a state of control, okay? Because just because you manufacture 3 process validation batches, and one of those process validation batch is at one extreme end of your allowable criteria, and the other one is at the other extreme end, that doesn't necessarily mean that your production process or whatever process you're validating is being maintained in a state of control, okay? It can mean that there's factors in there that could cause it on any given point in time to go out of control. Some common deficiencies found during inspections, all right? Process validation, all elements of process validation. I know when I go into a facility, one of the first things I like to see is process validation. And part of the reason behind that is I need to understand your process. I need to understand how you're making something, what you're making, what equipment you're using. And some of the best way for me to do that is to go in and look at your process validation. When an FDA investigator enters your facility, they have a very limited amount of time to understand your facility, your infrastructure, what you make, how it's made, and all the little intricacies of that process or of that product, okay? Think about how long you take to train a new employee in one area, okay? And then think the FDA investigator that comes in needs to have an understanding of that one area that you took however long it took to train that employee, days, weeks, or months, in a very, very condensed amount of time, okay? So some of the best ways for us to do this is look at process validation and equipment qualification. Okay, laboratory focal points. We like to look at OOS results. It gives us an idea of how your products are behaving, all right? It also gives us an idea of your investigative process. Do you thoroughly investigate an out of specification result? And are you able to determine a root cause? Is it a systemic problem? These are all things that we think about when we pull OOS investigations and we're screening OOS investigations that we're going to select for further review. We also like to make sure that your instruments are calibrated and qualified. And again, qualified for use, for what you're using them for, not qualified for something that is completely irrelevant to your facility. And I have seen this before. I have seen things qualified for a process that's not even part of what the facility is doing. But it was qualified for something and they perceived that to be okay. And so we said, no, this needs to be qualified for how you're using it, not just qualified for something. The analyst should be trained and have training that's documented, okay, so that we are able to look back at their training records and see that they have GMP training, training for the tests that they're performing, training for what they're reviewing. And just the normal stuff, like the everyday stuff, reference standards, reagents, samples, all of that stuff has to be properly stored. So typical housekeeping things, okay? It just keep a good laboratory, keep a good house. You know, if I walk into a laboratory, and I'll flat out tell you, if I walk in and I see a nicely organized laboratory, things are easy to read, stuff looks to be in date, things are properly stored, that already starts to give me a good feeling of at least, okay, this facility is organized, it appears to be clean, well kept, and it appears that attention to these kind of details is being made, all right? So that's like a very first impression of your quality system. It's kind of like walking into somebody's house for the very first time, okay? You walk in, and if it's a complete chaotic mess, it's like, ooh, what happened here? But if you walk in and it seems to be in an organized status, right? Maybe it's not perfect, but it's in an organized functional status. You're like, okay, this person, they live here, but you know, they seem to be an organized functioning person. All right, same thing with a laboratory. Okay, so compliance. All right, we already mentioned this at the beginning. It is a continuous obligation. All right, this is not a one-time achievement. It's not something that you can do one time, hey, great, we managed to stay compliant, the FDA came in, we're good, all right? No, this is something, like I said at the beginning, that you need to focus on literally all the time, all right? This is a continuous process. It needs to be built into the culture of the organization. Corrections to observations, when they're found during an inspection, need to be thought out and comprehensive, okay? You need to take into consideration any marketed product when you're thinking about these corrections and how that observation is affecting the marketed product, because that product is on the market being used by consumers. You need to address the impact that that observation has on the marketed product. Don't rush to initiate and complete a correction to try to get a deficiency that was found during the inspection removed from the 483. And I can tell you, I've seen this a number of times on inspections where the firm they're in such a hurry to try to correct it, to show us that they're going to be compliant. They're like, we're going to be compliant, we're correcting it right now. Where the correction ends up not being comprehensive enough and not being thorough enough. And what can happen if they're rushed through is down the road, if they have a problem or another investigator comes in and then finds a deeper problem with that same observation, now you have a much bigger problem, okay? So we appreciate the effort when you're showing us that you are going to make an effort to correct the observation. However, and I tell firms this all the time, when they come to me with an immediate CAPA, I'm like, I appreciate it. However, you need to think this through and make sure that you're covering all your bases on a correction, all right? Don't just rush through to try to get it done when the FDA is here, because it's on the 483, it's gonna stay there. It's not gonna affect things. But if you don't appropriately correct it, it's definitely gonna affect things moving forward. And I mean, it can affect things in a much worse way than just the FDA comes back and finds another observation, okay? You can end up and problems with marketed product, depending on what that observation was. So we're talking about FDA 483s. What is an FDA 483? Okay. It's an official document issued by the U.S. Food and Drug Administration at the conclusion of a facility inspection. So we're done spending our time at your facility. We found some problems that are not consistent with current GMPs. We wrote them down on a form, and we're going to issue this to you. Okay? It lists specific objectionable conditions or practices that we observed. These can be anything from we had a really bad rainy day, and the roof over your quality control lab on that particular day decided to give out, and there was water dripping from the ceiling over your HPLCs. To we found investigations that weren't complete. You know, the range is very vast, okay? But there are objectionable conditions that we found that your facility is not compliant with CGMPs. So what are the observations? They're factual findings, and they represent real risk, okay? What is real risk? Anybody in the room? Real risk. What is it? Is it a risk to a product? Who thinks it's a risk to a product? So I know you're all awake. Who thinks it's a risk to a patient? Who thinks it's a risk to both? Okay, it can be a risk to a lot of different elements, okay? But we like to look at risk. What is the higher risk? What is the risk of the deficiency that's been found, okay? Observations are written factually. They're not legal conclusions. It doesn't represent what the final determination of your firm will be. These are just observations that we found during the inspection, okay? A lot of firms will ask me when I present a 483, tell us what our major problems and our minor problems are, or which one of these are critical. What I can tell you, if it's on a 483, it's not minor. And you need to view the observations from the perspective of risk. That's how I view them. And if you're looking for a way, let's say you get a 10-item 483 and you're like, oh, which of these do we have to really address first, and then we'll work on the other ones later. Again, risk, what's on the market, what's more critical, what's higher impact. Those are the ways that you as a firm can streamline these. We do not, okay? A Form FDA 43 is a precursor to regulatory action. It's not immediate regulatory action. It's just telling you that you have problems that are violations of good manufacturing practices, current good manufacturing practices. If those problems are not resolved, then that can potentially, down the road, escalate to a warning letter or any other action that can be taken against you, okay? All observations, like what I just said, in our eyes, are created equal if they ended up on the 483, all right? Now, sometimes we'll list certain observations first versus last, and some of that is just prioritizing what we want to discuss first, all right? What is our first thing that we're thinking of? And then, you know, your inadequate investigation may be first, and your dripping ceiling may be last. Or if your dripping ceiling is dripping over a sterile line, that may be first, and your inadequate investigation may be last. But the thing is, they're all on a 483, and they all need to be reviewed equally and thoroughly corrected. The FDA doesn't have a formal severity ranking for observations placed on the 483. We just put them on the 483, and they are identified as CGMP violations that need to be addressed, okay? So they're all important. Your firm bears the responsibility of doing a risk assessment to determine which of those are the higher risk, which ones hold a heavier risk, and like I said before, if you have to choose an order of correction, which one do you want to correct first? Or which one do you want to put the most effort into first, and then show us that, yes, we're putting effort into the other 5 or the other 2, but it's a little bit more gradual, okay? All observations have to be included in your response with an adequate level of tension and urgency, okay? If you respond to an observation with rhetoric that reads as though you perceive it to be minor or not important, that can be a red flag to us when we're reviewing what your corrections are going to be. So evaluating deficiency risk to product quality and manufacturing. Does the deficiency identified have a direct or substantial potential to impact product safety? That is a big question, all right? That is a question I ask myself probably 100 times when I'm at a firm, looking at their data, looking at their process, and looking at what they're manufacturing. Is the deficiency found to have occurred in an area that has direct proximity to a manufacturing process? Like I just mentioned, a leaking ceiling over a filling line, okay? Yes, that would have direct product impact, okay? So we think about these things when we're generating these observations. When you're writing your corrections, try to think in a similar manner. If you were to find that observation, what would you look at? All right? Okay, yes, this is a problem. It's right over a manufacturing line. Or maybe we found a leak and there's a problem in your ceiling, but it's off in a corner of your warehouse where it would have to become substantial to get to a product. And you can say, okay, you know what? We're going to address correcting this other observation, and we'll address this observation, but we're going to take care of this more important one, and then we're equally going to give time and attention to the leaking ceiling. We're not going to short it, but this is how we're perceiving this, okay? Does that make sense? We look at risk. So ultimate question, what is the risk? Risk is probability of occurrence of harm and severity of that harm. So when we're looking at risk, we're like, what is the potential that we could harm a patient and to what severity? So on the outside of the FDA, my other life, I was a practicing pharmacist, okay? And when I look at things, I know patient populations and things that certain medications are given to. I do think, what is the risk to this patient population? If I were still practicing pharmacy, or if I were practicing pharmacy and you handed me this drug product from this facility, knowing what I know, sitting in that facility on that day, would I be comfortable handing that product over to a patient or over to a provider to administer to a patient? And that thought does go through my head every time I'm at a drug manufacturing facility. So quality risk management is a valuable component, and it applies to developing an effective quality management system. And like we were talking before, when addressing observations, you want to look at patient safety, product safety, and quality. You want to make sure you conduct a comprehensive risk assessment. Design your corrective actions to be applicable to the observation and comprehensive to the observation. So how do you respond to this document that I just handed you? We have a handy dandy electronic submission process now that you will receive the instructions when you receive the FDA 43. All right, it's an electronic set of instructions, and all responses now go to an email address. Firms still have 15 business days to respond to the 483. And when you receive this electronic submission instruction handout, if you have questions while the investigator is there, please ask. And we're more than happy to answer your questions and help you work through submitting your response. Okay, so the response submission method. Electronic submission only, we're not mailing things anymore. We've stopped killing trees, we've stopped going overboard on postage stamps. Now everything is going through the electronic waves. There is a file size, it's 100 megabytes per email. Sometimes that file size changes, so you can check and see if the file size, the maximum file size you can send in that email has changed, okay? I remember it used to be a little bit smaller. It's gone up recently, but you want to verify what's the biggest file that we can send per email. Sometimes responding might require 2, 3, 20, 30 emails. And then the subject line is applicable to the name of the firm, the FEI number. This is all in your electronic submission instructions, okay? And these can be found on that handout. Okay. What do you avoid in a response? Vague or generic responses that lack understanding of the observation. If you do not understand the observation, ask, please. We're more than happy to discuss it, answer questions, and help you understand what the observation is to give you the best chance at adequately correcting it. Because we want you to correct things. We don't want things to be vague and uncomprehendable. We want things to be fixed and to be good, and for you to have a good manufacturing practice. Don't set timelines that are not achievable, okay? Set realistic goals. Like I said, we're not in a hurry. We want comprehensive responses that make sense and cover all your bases. Okay, so that's my little spiel on inspections and 483s. So now let's talk a little bit about emerging technology. As regulatory expectations evolve, manufacturing complexity increases. There are new emerging technologies that can help you maintain a good quality system, okay? I've heard a lot of talk around about different technology systems. We're just gonna go through a couple really quickly, and I will give you one common theme that I look for when I go to a facility and I find a new piece of technology. First of all, I love it when they explain it to me because I see new things all the time, and I've seen some really neat new technology that I didn't even know was out there until I saw a firm using it. And I thought, wow, this is amazing. But then I have follow-up questions to that, okay? And they're always the same. So the implementation of new technology like AI, Internet of Things, you know, all these electronic systems that are being developed can introduce other risks. They can eliminate some risks, but they can give you other risks, okay? New risks that you have to take into consideration. So artificial intelligence and machine learning, these are two that are evolving. They can help monitor your facility, all right? Artificial intelligence can be used, we'd be here all day if we came up with ways that AI can be used. I hear something new every day about how AI can be used. It's pretty incredible. But these new technologies can be used to help you stay compliant. They can monitor things within your building, within your process, within your line, within your material, your personnel. There's almost limitless possibilities. So the Internet of Things, this is kind of neat. I've seen this connected at a few places. It has sensors and software and technologies that connect and exchange data. And it's kind of like Alexa at home, where you say, Alexa, turn on my lights. Alexa, shut off the toaster. Well, it's that to a massive scale. And I've seen some really cool uses of this, okay? Continuous environmental monitoring is one of them, where it can monitor multiple places at one time and send alerts in a very, very quick response time. If those places, if the room goes out of temperature, if the chamber goes out of temperature, and everything from robotic forklifts to robots that decarton vials and sterile manufacturing processes using a robotic arm to prevent operators from having to open the doors, okay? And that's a very cool process too. But one of the things that I look at whenever I see emerging technology, validation and qualification. Remember at the beginning of this talk, I said process validation, equipment qualification. Is that piece of technology validated for its appropriate use to show that it's reliable, generating appropriate data, and functioning as expected? And then I look at the data integrity of whatever is being generated. Can that data be manipulated? Can that data be lost? What happens with that data? Who reviews it? How is it handled? How is it used? I look at all of these things. And these concepts are applicable to just about every piece of new technology I look at, regardless of what the technology is, okay? So it can be the greatest thing in the world, and I'll go in and I'll think, wow, this is amazing. I actually saw something that was really cool over in Portugal, I don't know, a couple years ago. I was like, this is so cool. And at the end of the day, I'm like, how did you validate it? Because it was that complex. And it was kind of neat to see how they validated it, because it also gave me a further understanding of how it worked. But these are things to think about. Make sure your validation is comprehensive, and that whatever data you're generating is secure. Okay, so in our 28 minutes and 30 seconds so far, we've talked about compliance being built into the culture of your organization and not just a one-time point accomplishment. We've talked about addressing observations adequately. And we've talked about emerging technology that is very useful in a lot of situations, but also needs to be handled appropriately, risks identified with that technology, and then appropriate validation, data storing, data review being conducted to maintain CGMPs at your facility. Questions? Yes.
Can you come to the mic, please?
Yeah, come to the mic. I can hear you, but the individuals who are online may not be able to hear you, and some of them may have the same question.
Thanks for the talk. It's really informative. My question is more addressed to the electronic submission.
Yeah.
So I was thinking in my mind because I use the ECSG portal for in my submissions for INDs. Is that something that you're looking at converting over for your 483? So that way you have for that one facility, everything that's involved, all the inspections, not only for the FDA, but for personnel changes, right?
Yeah.
And so that one person who has it, they have that history instead of somebody's email.
You mean from the FDA perspective? Are we looking at converting to that?
Yes.
That question is way out of my lane. But what I can tell you is right now we have our 483 maintenance systems and we have our electronic submission systems. So we have different systems. Now, as an organization, for you as a firm to put all of that into one system, I could see where that would be very beneficial for you. Okay, that would be on your end to design and maintain that, but I can see how that would be very, very helpful from a lot of angles. Okay, but as far as what we're doing, I have not heard anything like that from the FDA. Okay. Yeah.
So excellent. Thank you so much. So now that companies are putting a lot of information data on the cloud, have you seen problems with that? How do you feel about that?
The data needs to be backed up. The cloud to me is, so I'm Generation X, all right? When I went to school, we used chalkboards. I didn't get my first personal computer until I was in professional school. So to me, the cloud is like this nebulous thing that came out of Star Trek that I'm still wrapping my head around, but I think it's fascinating and I think it's great. And I store a lot of my own stuff on the cloud. And I see definitely it's very useful for data storage. That being said, I would still say backing up your data to a reliable source where if the cloud vanishes tomorrow, you're able to recover your data. Okay, that is the part of the cloud that makes me nervous. And I see a lot of places storing stuff on the cloud, but I also see those same places backing it up in some degree to a known source that is then kept offsite in a fireproof box or in a central location. so that they're able to retrieve their data should the cloud fizzle out.
Excellent. Any more in-house questions, please? Please come to the mic, nearest to you. You're getting quite a lot of in-house attention here.
Hello.
Hi.
My question might be a bit pokey of a subject, but with the new one-day inspections, is there a checklist that you are going down that, let's say you don't find anything, are you up and leaving, or do you continue through with that inspection for the rest of the period?
So it's really coincidental and appropriate that you would ask me that, because I am on the one-day inspection pilot, about to do my first one-day inspection as part of that pilot, coming up pretty quick here. And I think it's a very interesting program. I'm kind of very excited to go out and do this, to see how it works and to see how we can apply it and how we can make it work and what the limitations are going to be and what the advantages are going to be. So I'm very excited about this because it's a cool new thing and I'm super excited to be on it. Does it mean that once we go through that checklist and don't find anything that we're going to keep going? So as an investigator, if I had a good feeling after completing that checklist that everything I looked at on that checklist was comprehensive, well done, and you had good housekeeping, would I feel okay closing out the investigation? Yeah, probably. However, if I saw something anywhere in a document, on the floor, anywhere going through that facility that would put question into my head that even though what I was looking at seemed okay, that I had a concern, I would pursue that concern even after that checklist is over. And that's just for the sake of being comprehensive, okay? Does that mean it's going to result in anything? No, but if I have a concern, I will still pursue it. And if it goes into more than one day, I've done my best and we'll just go into more than one day. But, I would like to try to stay to that. And it doesn't, just because it's a one-day inspection doesn't mean you're not going to get a 483 observation. I mean, there are 483 observations being generated on one-day inspections. So it doesn't eliminate the possibility of a 483 observation. We're just trying to streamline things a little bit to be a little bit more efficient, and we're going to see how it works. But I think it's a very cool pilot. Like I said, I'm very, very excited over this.
Thank you for that. Do we have any online questions?
Sure, go ahead.
Can we take an online question?
Oh yeah, let's do an online question, then we'll do her question.
Of course, thank you for that. So one question says, in writing a response to a 483 observation, how do you determine what is a reasonable amount of time to make the correction on the part of the firm?
So it's what is the correction that has to be made? What does it involve? And how feasible does it look? And if you're the firm estimating that, and let's say you're 3/4 of the way to completing your correction, and you realize the deadline is tomorrow, email whoever you're corresponding with regarding the 483 and your observations and your responses to those observations, and let them know and extend that timeline and state why. I've seen this done before. I've seen CAPAs extended. I've seen timelines extended. And the CAPAs that I've reviewed, when they've been extended for a reasonable reason, or something that makes sense, or something that's like, I look at it and I say, yeah, you know what, I understand why you couldn't get that done within that time frame. I completely understand why I had to extend that CAPA. And if they're going on the same theory of we're going to be comprehensive, we're extending the timeline to be able to include these things and appropriately do this. As an investigator, when I look at that at a firm, I don't get all kinds of crazy. I see CAPAs extended all the time. It's what is your reason for extending it?
Thank you very much. We have one more question in-house.
Yeah, thank you so much for this. I'm wondering, so from the perspective of a company that is developing products and sponsoring clinical trials and building that supply chain for API manufacture, I'm wondering how we have access to understanding the CGMP compliance of potential CMOs, contractors. Is the 483 public or only when there are warnings? Is there some way to get that information?
So there's a lot of ways you can go. You can actually go onto the FDA website, and there are published warning letters on there. And you can go and look and see if they had a warning letter at any point. You can request from them their inspectional history, and you can request certain data from them. You can sometimes within the public library of the FDA. So we have a library page where we release documents. Sometimes on that library page, you'll find 483s that have been redacted and approved for public release. That's another place that you can look. But you can ask them and see if they'll give that to you. You know, ask them if they've had any 483s and ask them for any EIRs or inspectional history that they may have had.
Thank you so much. This is really helpful.
Oh, you're welcome.
Thank you very much, Commander Shields. And let's give Commander Shields. It is my pleasure now to welcome Dr. Grandinetti for her talk on achieving quality through purpose-driven clinical trials. Thank you.
Good afternoon, everyone. Okay. So today I'm going to talk about FDA's perspective on ICH E6R3 and achieving clinical trial quality through purpose-driven clinical trials. But I'm going to tackle it from a slightly different angle. So I'm curious, how many of you in this room have seen The Bear, which is a series on Hulu? Oh, good. At least a few of you. Well, a few months ago, I rewatched all 4 seasons of The Bear, and I caught myself thinking, this is actually a pretty good analogy for what ICH E6R3 is trying to achieve with quality by design and its risk-based framework. And the more I watched, the more the parallels started to click. So for those of you who haven't seen it, here's the quick version. It follows Carmi Berzato, who's a chef trained in Michelin-starred restaurants, who returns home to Chicago after inheriting his family's sandwich shop, which is called The Original Beef. The shop is popular and functional, but it runs on chaos. The staff rely on habits, shortcuts, and constant firefighting rather than any proactive or structured approach. Over the course of the series, Carmi gradually transforms and redesigns the sandwich shop, introducing structure where it matters most, focusing the team on what's critical, allowing flexibility in areas of lower criticality, and making the whole system more intentional and consistent. So spoiler alert, so cover your ears if you haven't seen it and you don't want to know this. By the end of the 4 seasons, he has evolved the restaurant from a reactive neighborhood sandwich shop into a disciplined, high-performing restaurant operating at a Michelin caliber level. So today we're going to look at clinical trial quality and implementation of E6R3 through the lens of Carmi's transformation of The Original Beef to The Bear. So first, I'm going to talk about FDA's role in the development of E6R3 and its adoption and implementation in the U.S. I'll discuss E6R3's risk-based framework and those foundational principles that make up that framework. And then I'll talk about some implementation challenges we're seeing and what it actually takes to make it work. And then finally, I'll talk about what FDA and our international regulatory counterparts are doing to support this shift. So let me start with FDA's adoption and implementation of ICH E6R3. So first, the guideline update was something that FDA actively supported, recognizing the need to evolve GCP to better support innovation while allowing for more flexible, fit-for-purpose approaches to clinical trials. FDA played a central role in developing this guideline. We served as the rapporteur on the ICH E6R3 expert working group. And in September of 2025, we issued E6R3 as FDA guidance. And I want to note that while E6R3 is issued in the US as guidance rather than regulation, we view it as the gold standard for clinical trial conduct. And it clearly communicates our expectations for how trials should be conducted. It's what we expect to see in practice and what we'll be looking for in both our inspections and in our clinical trial reviews. And looking ahead, you should expect future updates, whether guidance documents, inspection approaches, or policy decisions to continue to align with E6R3's GCP principles and annexes and to support risk-based approaches and innovation in clinical trials. And speaking of where we've been heading, over the past few years, we've published several guidances that already align closely with E6R3. Together, these guidances reflect FDA's broader push towards more flexible and efficient clinical trial approaches, including trials incorporating decentralized and pragmatic elements, digital health technologies, and adaptive designs. Many of the guidances shown on this slide reinforce those same principles in E6R3, encouraging quality by design and risk-based approaches. So on this slide, you'll find links to the comprehensive ICH training available for anyone that's interested in E6R3. ICH contracted with the Multi-Regional Clinical Trials Center of Brigham and Women's Hospital in Harvard to develop this training. But I also want to give a shout out to the ICH E6R3 Expert Working Group, who has also been actively involved in shaping and developing the training content alongside MRCT. And I'm very proud of what's been put together here. And I encourage you to take a look if you haven't already. As shown on the slide, modules 1 and 4 are already published, and they cover the foundational principles and informed consent. The remaining modules, 2, 3, and 5, will be released soon, and they'll be covering responsibilities and oversight, data governance, and essential records. So let's talk about how we got here, because I think understanding the evolution of clinical trial quality helps explain why E6R3 is so important. So historically, quality in clinical trials was defined by what I call checkbox compliance. Traditional quality management tends to take a one-size-fits-all approach, applying the same level of scrutiny and effort across all data, systems, and processes, regardless of their relative impact or importance within the trial. The underlying assumption was more monitoring and source data verification equals better quality. But we've learned that this approach doesn't actually deliver meaningful quality. It's resource intensive, it's inefficient, and it doesn't ensure that the most important trial attributes and risks are being addressed. So you can check every box and verify every data point and still miss the issues that matter most. So not all data, systems, and processes are equally important, and focusing on the wrong things can actually reduce quality by diverting your attention and resources away from what truly matters. And so the shift now is to take a more deliberate and thoughtful approach to trial design and conduct. Adopt focus management and oversight where it matters most. And E6R3 and E8R1 are our roadmaps for this. So the purpose of the E6R3 update really comes down to making the guideline more usable in today's clinical trial environment. E6R3 is designed to facilitate modernization, harmonization, and innovation in trial design and conduct. It addresses the increasing complexity of global clinical trials while still remaining flexible enough to support a wide range of trial designs and operational approaches. At its core, it promotes a risk-based proportionate approach, and it emphasizes aligning management and oversight with the critical to quality factors and the level and type of risk, rather than applying a uniform approach across all types of trials and trial-related activities. It strengthens our expectations for proactive risk management across the trial lifecycle. And then that same risk-based approach extends to data governance, which is an entirely new section in this guideline. And there are also additional enhancements, like, for example, it allows for more flexibility in informed consent to support remote and electronic informed consent, and to also accommodate patient preferences. So let's talk a little more in detail about 3 of the 11 foundational principles in E6R3. And these 3 principles really serve as key pillars of that risk-based framework. And they're quality by design, risk proportionality, and fit-for-purpose clinical trial quality. And again, in the BEAR, you see elements of all 3 of these principles in action, just in a very different setting. So the first principle that I want to highlight is Principle 9, which talks about the quality of a clinical trial being fit for purpose. And at its core, the principle recognizes that quality isn't about data perfection. It's about whether the trial is designed and managed to meet its intended objectives and is of sufficient quality to provide confidence in the trial results and support sound decision-making. And this is all while protecting participants. And while some errors may occur in a trial, the focus really is on preventing those that can meaningfully affect participant protection or compromise the reliability of the trial results. And so again, this means moving away from one-size-fits-all quality standard towards quality approaches tailored to the trial's specific objectives and design. And that's an important mindset shift. And the shift is empowering because it allows us to work more strategically about ensuring clinical trial quality rather than chasing data perfection. The second principle that I want to highlight is quality by design. And this is in Principle 6 in E6R3. And this certainly isn't a new concept. We've been talking about it for years, and it's now reinforced in ICH guidelines, particularly in ICH E8R1 and now in E6R3. And quality by design starts at the design phase, and it's not something that you can retrofit or evaluate after the fact. And as Carmi transforms the original beef into the bear, he starts at the design phase. He refines the menu to align with the restaurant's vision. He restructures the kitchen. He defines roles and responsibilities. He sets standards and establishes a system that's proactive to issues rather than reactive. And that's really the essence of quality by design, designing your trial from the outset to meet your objectives and reliably answer your research question, identifying those critical to quality factors early, and then building the trial around them to protect and ensure their integrity. And we're going to talk more about critical to quality factors on some upcoming slides. And while quality by design starts during protocol development, it doesn't stop there. It really sets the foundation for an ongoing adaptive quality management approach throughout the trial lifecycle. And as issues come up, you refine your mitigation strategies based on what you learn so the system can adapt while still keeping control of and protecting the trial's critical to quality factors. And then the third principle that I want to mention is risk proportionality, which is highlighted in Principle 7 in E6R3. And risk proportionality is inherent in the quality by design approach, and it ensures that trial management, oversight, and quality efforts are aligned with the level of risk to your critical to quality factors. So simply put, areas of higher criticality receive more attention and resources, and areas of lower criticality receive less. And this is not about doing less, but doing the right work in the right places. So back to the bear. We see both sides of this concept in season 2 on opening night of the new restaurant, where Carmi gets unexpectedly locked in the walk-in freezer because of a broken freezer door handle. Before getting locked in the fridge, he demonstrates really a complete failure of risk proportionality because he panics over the 7 fishes dishes that's sitting on the counter. He starts aggressively screaming at his sous chef that the plates have gone cold and that they need to fire the plates. So he treats this localized kitchen delay like an apocalyptic emergency, all while completely ignoring the major operational hazard that's been on his to-do list for weeks, which was calling a repairman to fix the broken freezer door. So risk proportionality is about your ability to appropriately or proportionately match the intensity of your response or mitigation strategy to the severity of the risk to the critical to quality factor, rather than placing a disproportionate high-intensity response to minor issues or underestimating serious ones. So once Carmi is trapped inside the freezer, the team takes over and models true risk proportionality. They don't waste energy trying to fix every minor mistake. Instead, the team is able to adapt, they redistribute responsibilities, and they focus and consciously align their resources with the critical to quality factors, concentrating 100% of their efforts on what most directly impacts whether the evening succeeds or fails. And that's coordinating tickets and dishes, maintaining kitchen flow, and getting food to guests. So in the bear, the quality they achieve on opening night is not flawless, but it is sufficient to deliver the intended outcome, which was a completed service that was sufficient for the guests to leave the restaurant satisfied. So, and it's important to recognize that quality by design, the identification of critical to quality factors and their associated risk, and quality management are not separate concepts. They are interdependent, and they need to work together within a risk-based framework. So you can't just cherry pick pieces of the framework and expect it to function properly. And when these elements are aligned and implemented effectively, the result is fit for purpose clinical trial quality. And that's what E6R3 is asking us to achieve, not data perfection, not more work, but the right work focused where it matters most. So this brings us to critical to quality factors. And it's important to spend time on this because these factors are often misunderstood. We want sponsors and investigators to avoid errors that matter. And the errors that matter are those that impacting the trial's critical to quality factors. So critical to quality factors represent attributes of a trial that, if compromised, can impact participant protections. It can lead to results we can't trust, or worse, decisions that might not be ethical or safe. Critical to quality factors should be individualized for the trial, meaning that while some critical to quality factors may appear consistently across trials, their relevance and importance should be evaluated on a trial-by-trial basis. And critical to quality factors need to be considered holistically so that dependencies among them can be identified. Sponsors should also engage in multidisciplinary, cross-functional discussions within the sponsor organization with their service providers and with external parties, such as investigators, patient advocacy groups, and patients. These conversations really bring together scientific, operational, and clinical perspectives to critically assess and identify what matters most in each specific trial. So this slide shows examples of areas in a trial where critical to quality factors may be identified. And many of you may recognize this because it is a small excerpt from the Clinical Trials Transformation Initiative Quality by Design Principles document that they developed over 10 years ago. And it's not intended to be used as a checklist or even as an exhaustive list, but it's intended to be used as a way to prompt discussions to identify potential critical to quality factors and support conducting robust risk assessments. So I'm going to use another example from the bear. In the first few episodes, the Italian beef sandwich becomes a central focus. Because critical to quality factors should be identified holistically, so not just looking at the final product in isolation, let's use the Italian beef sandwich as a critical to quality factor, and let's identify several supporting dependencies or other critical to quality factors that contribute to its overall quality. So, for example, within that Italian beef sandwich, the type and cut of beef are critical factors. The beef also needs to be properly cooked, sliced the right way, and held at the right temperature to maintain consistency and quality of the beef. And the bread needs to be made correctly. And if the beef sits too long, or if the sandwich is assembled incorrectly, or the bread gets soggy, then the sandwich fails, even if the beef itself is good. So the cooking and preparation process is also critical to quality. And so the key point that I'm trying to make with this example is that critical to quality factors, again, aren't just the final product in isolation. They're also the critical inputs and the steps that determine whether the outcome actually works. So if we apply the same critical thinking to a clinical trial, for example, we'll take a primary efficacy endpoint measure that drives the trial's conclusions. So this is an easy critical to quality factor to identify. But getting reliable endpoint data isn't just about what you measure. It's about how the systems and processes around that measurement are designed and managed to protect the quality and integrity of that endpoint data. And these critical processes and systems are not always so easy to identify. So it's important to map out the data flow and understand the technology used in the trial, including the data integrity and security controls, understand the data collection processes and procedures, and understand the inherent risk to those critical to quality factors. And getting this right and truly understanding the critical to quality factors and their associated risk in the trial is the foundation of quality by design and risk proportionality. So let's talk about the reality of the implementation of this risk-based framework, because this is where it sometimes gets hard. There were 2 surveys done in 2024 that really helped shed some light on this. One was from the Tufts Center for the Study of Drug Development with 206 respondents across 125 companies. And another was from the Association of Clinical Research Organizations, looking at about 5,000 trials across 7 CROs. And in general, 3 issues stand out in these surveys: knowledge and understanding gaps, fragmented implementation of those foundational principles, and cultural barriers. So first, there's a gap between perceived understanding and practical application. Based on the survey data, many people appear to be familiar with quality by design and quality management in theory, but that knowledge often isn't deep enough to guide real decisions in trials. So while the concepts are recognized, they're not always fully internalized or applied effectively. Second, implementation tends to be fragmented rather than holistic. Organizations are adopting pieces of quality by design and quality management, but not operating within the fully integrated framework. So we're seeing risk assessments being performed, but they don't reliably drive mitigation strategies. And processes like monitoring and risk review don't consistently connect into a feedback loop. And this often results in a checklist style linear approach of plan, conduct, and detect rather than a dynamic, adaptable, risk-informed system. And then third, culture plays a major role. While there's general agreement that risk-based quality management can improve quality, there's less conviction around its operational benefits. And that makes it harder to sustain commitment. And it seems that many organizations are still operating with a quality by inspection mindset, focusing on finding issues after they occur instead of preventing them up front. So the cultural challenge is the most fundamental. Putting quality by design into practice means really rethinking how we approach clinical trial quality. And again, it's shifting from a checkbox compliance and quality by inspection to building quality in from the start. It's moving from siloed functions to true cross-functional alignment around the critical to quality factors. And it's moving from static plans to an ongoing data-driven process that evolves with what we learned. And these shifts are difficult. They require changes in mindset, and they require organizations to rethink how they are structured and how resources are allocated. And I know you're going to get tired of these bear references, but I got a few more, so bear with me. So the bear really does an excellent job illustrating how hard the cultural mindset change is. So when Carmi begins transforming the restaurant, the team doesn't immediately say, great idea, chef. They're resistant, they're skeptical, and they're deeply uncomfortable with the uncertainty. So Richie, who's Carmi's cousin and the floor manager, has been doing things exactly the same way for years, and he pushes back against every change. Marcus, who's a pastry chef, is worried about whether he can even operate at that elite level. And then Tina, who's a line cook, is terrified about whether she still has a place in this new kitchen. And this is exactly what cultural change looks like. It's rarely a resistance to quality itself. It's a resistance to uncertainty. It's a concern about identity, competence, and belonging in a different system. And we've operated for decades in a clinical trial world where quality has been associated with applying the exact same level of scrutiny to everything, regardless of the criticality of the data, the systems, or the process. Entire businesses and systems have been built around that comprehensive model. So even though the conversation around quality by design began in the early 2000s and is now deeply embedded in E6R3, the shift feels a little more fundamental than an incremental update. And that creates some very real questions like, what if we miss something? What if reducing oversight creates a blind spot? What if regulators expect something different? And that's exactly where Richie was early on in the show. He wasn't resisting because he didn't care about quality. He was resisting because he wasn't confident yet in a different definition of quality. But then Carmi sends him to train at Ever, which is one of the best restaurants in the world, and something shifts. A light bulb goes off for him while he's performing one of the most basic tasks, which was polishing forks. At first, he feels it is completely insignificant, but then he realizes that the fork is a guest's very first interaction with the restaurant. And if it isn't right, then the integrity of the entire experience is already compromised. So he starts doing it differently with purpose and intention because he finally understands the purpose behind the critical process. And that's the cultural mindset shift that E6R3 requires. It's not about caring less about quality. It's about being more intentional with it. It's about focusing on what truly matters, building quality into the process from the start rather than trying to inspect it in later. And importantly, everyone understanding and aligning on what the critical to quality factors are in the trial and how their role impacts the critical to quality factors or is important in protecting those critical to quality factors. One more important roadblock that I want to mention is that sponsors and investigators have expressed reluctance to adopt risk-based approaches for fear that inspections may not align with the risk-based framework. This hesitation really produces overly cautious, process-heavy practices that undermine the efficiencies and quality gains that E6R3 seeks to achieve. And a risk-based inspection approach helps remove this barrier, and we're working towards aligning our inspection focus with E6R3's risk-based framework. And this alignment should give sponsors and investigators confidence to apply the framework without fear of punitive findings. And my message here today is that thoughtful, risk-proportionate practices are both expected and accepted. FDA has been actively collaborating with other regulatory authorities for more than a decade, including the EMA, PMDA, MHRA, and Health Canada. These partnerships help improve inspection coverage. They reduce unnecessary duplication of effort and enable the sharing of inspection findings. They also deepen our mutual understanding of each other's processes, and they support joint inspections when appropriate. And a key part of this international collaboration is the Pharmaceutical Inspection Cooperation Scheme, or PICS. And PICS is a non-binding partnership of 56 regulatory authorities around the world with the mission of harmonizing inspection standards globally. And in 2022, PICS established the expert circle dedicated specifically to GCP inspections. So the GCP expert circle focuses on developing training and practical guidance for GCP inspectors, promoting a consistent and harmonized approach and aligning the inspection practices with the E6R3's risk-based framework. The PICS GCP expert circle held its first training meeting in November of 2025, bringing together more than 160 GCP inspectors from around the world. And the training emphasized consistent interpretation of E6R3 with a strong focus on its risk-based approach. A key theme was ensuring inspection activities are centered around the trial's critical to quality factors and whether sponsors have clearly identified those factors, assessed the associated risks, and designed their trials and quality systems to appropriately protect them. So let me bring this all together into some key takeaways. So first, E6R3 isn't just an updated guideline. It's a fundamental shift in how we think about what quality looks like in a clinical trial. Second, this transformation is built on some foundational principles, quality by design, risk proportionality, and fit-for-purpose clinical trial quality. Third, implementation of the risk-based framework is challenging. It requires both policy alignment and a cultural shift. You can't just update your SOPs and call it done. You really need people to understand and embrace the philosophy behind quality by design and risk proportionality. Fourth, regulators are committed to supporting this shift. We're aligning our inspection practices, we're collaborating internationally, and we're providing training and resources. And we're on this journey with you, learning and evolving together. And finally, I want to close with a moment from The Bear, Season 2, that really beautifully reflects, I think, the essence of quality by design. So when Carmi sends Richie to train at Ever, Richie notices a sign in the kitchen that reads, "Every second counts." And at first, he interprets it as a demand for relentless speed and perfection, and even the smallest of tasks. But as he becomes immersed in the kitchen culture, he realizes that it means something deeper. It's not about speed and perfection for its own sake. It's about intention and purpose, and a way of working where decisions, details, and processes are intentionally designed and aligned around a clear purpose. What is critical for success? And in that restaurant, it was about creating the best possible experience for the person being served. And that is exactly where we are with E6R3. We're moving beyond viewing quality as a checklist of compliance requirements. Instead, we're embracing a mindset where quality is intentionally designed into the trial, guided by the trial's specific objectives, the critical to quality factors, and the risks that can threaten their integrity, grounded in purpose, and focused on what truly matters for each individual study. E6R3 and E8R1 gives us a framework, and now it comes down to you. So I'm happy to take questions now, and I promise I won't use any more Bear references.
Thank you, Cheryl. Actually, we got a couple comments online saying how much they appreciated your Bear references.
Oh, thank you. That's good. It was sort of a gamble.
Any in-house questions, please? I guess we're setting the mood here with the lights going down.
Yeah, first of all, thank you very much for the talk. I really enjoy. And I would like to take a quick survey here, like how many from all of you coming from the sponsor side? All right. And how many from all of you doing the clinical trials with the insurance that cover all the AE and SAE rather than only the related AE/SAE? All right. All right. So that's related to my question. So I believe that patient is the first stakeholder in any clinical trials. And then I just checked the ICH GCP E6R3 and the section 3.14 about the insurance. And this insurance topic is always become questioned in my country during the inspection from the FDA. And then about the ICH R3, it mentioned that the guidance not specifically required us to cover which AE or SAE, but related to the applicable regulatory in each country and each land. And my question is, since I'm here, I would like to learn from the perspective of the FDA, what kind of insurance that actually being like the hope from the FDA that is covered? Is it like all related to drug only or all the AE/SAE? Because if it's like 10 years clinical trial, from the sponsor point of view, it will be like not cost effective. And yeah, I would like to have this.
Okay, I'm a little bit confused. When you say insurance, I'm not sure I know what you're talking about.
Oh, it's a clinical trial insurance.
And you said that's in 314? Can you read the actual provision for me? Because I'm sorry, I'm not familiar with that.
This one. So in the section of 314, insurance or indemnification compensation to participants and investigators. If required by the applicable regulatory requirements, the sponsor should provide insurance or should indemnify legal and financial coverage, the investigator or the institution against claim arising from the trial, except for claims that arise from malpractice and or negligence. And then the second one, the sponsor's policies and procedures should address the cost of treatment of trial participants in the event of trial-related injuries in accordance with the applicable regulatory requirements.
Yeah, I'm going to have to pass on that question. I'm sorry. Maybe we can talk offline.
Okay. Yeah. Thank you.
Sorry about that.
Any other in-house question, please? Corey, do we have any online questions?
We do. The first question here is, if we shift from doing a 100% data verification to focusing only on critical to quality Q or CTQ factors, how do we prove to an auditor that the data we didn't verify wasn't actually important?
Okay, so that's a really great question. And so what I would say is that you really need to document your risk assessments. You need to document what you identified as critical to quality and how you control those. And you need to provide your rationale. So, you know, if you have all of that documented and you have it, and it's you have good sound rationale for what you've done, then, you know, I think that that would be the first step. You know, oftentimes we do see companies not identifying something that was critical that turned out to be critical in the end. But again, remember that you're constantly monitoring these critical to quality factors. You're also reviewing your mitigation strategies to make sure they're effective and you know, in protecting them. And then as the trial goes on, you might identify more and critical to quality factors, so then you have to adapt. So we want to see that entire process. And there was one thing about that question that I didn't quite like, because you said only, you know, only monitor CTQ factors. That's not what we're saying. We're saying that you focus your resources, you prioritize these CTQ factors, but that doesn't mean you ignore everything else in the trial. You're determining what level of resources and what level of oversight and management you need to give. And some get more and some get less. So you have a primary efficacy endpoint, you want to make sure that's right. But maybe you have an exploratory endpoint that you don't need to monitor as frequently. You can check it, every so often, or check only a certain percentage of that data. So that's what that means. It doesn't mean that you ignore everything else in the trial. You still have to check everything, but you're going to put more resources into ensuring that you're getting what's critical correct.
Thank you for that clarification, Cheryl. Any in-house questions? So I think we have time for one more online question, Corey, if we do, please.
Sure. So we have one more online question that reads, the FDA guidances are named guidance for industry. Do they also apply to non-commercial sponsor investigators?
They would, so the guidances guidances are non-binding, first of all. They're not regulation. So they tell what FDA's current thinking is about a topic. In terms of non-commercial sponsors, we shouldn't have two standards for clinical trials. So ICH E6 R3 applies to trial sponsors that are going to submit to regulatory authorities, but it also applies to academia because a lot of those trials may get submitted. They may not have that in mind when they're doing the trial, but later often do get submitted to the regulatory agency to support a regulatory decision. So again, do they apply or not? I think the guidances tell our current thinking, and you should follow them. I guess that's all I could say about that.
Thank you very much. Thank you.
Okay.
A short break now. We will reconvene at 3:05 p.m. Thank you.
Mic check.
We'll be having our one-to-one sessions 4:35, beginning 4:35 and ending at 4:50, at which point we will then adjourn for the day. But let me take the honor to introduce our next two speakers. First, we'll have Annie Saha, who is the Associate Director for Data Science and AI Policy at the Office of Medical Policy. Annie is acting in this role, and she will speak to us about the use of AI to advance drug development. Then Annie will be followed by Dr. Oanh Dan, who is the Senior Pharmacist with the Regulatory Science and Applied Research Lead. She is the Regulatory Science and Applied Research Lead, CDER Emerging Drug Safety Technology Co-Lead of the Regulatory Science staff at the Office of Surveillance and Epidemiology within CDER. And Dr. Dan will be speaking to us about FDA CDER Emerging Drug Safety Technology Program. And, we've been talking about AI all day long, right? So I know everybody's been waiting for this. And we have also mentioned emerging drug safety technology in quite a few lectures so far. So let me please welcome Annie Saha to the podium.
Right, let's see. All right, everyone good on mic check? Cool. All right. Okay. All right, so I'm going to talk about AI to support regulatory decision-making for drugs and biologics. And along with my acting role in CDER, my normal day job is that I'm Associate Director for Strategic Initiatives in the Digital Health Center of Excellence on the device side. So I'm sure, as everyone in the room knows, AI is being used across the drug product lifecycle. We really see that AI has the opportunity to reduce the time and cost of drug development. And some of that is outside of FDA's scope in terms of what we would look at, but we really do encourage where AI can really bend the cost curve and help get treatments to patients sooner. And so we really see the use of AI across the entire life cycle when you talk about in the discovery phase, so drug target identification, selection, prioritization, compound screening and design in the non-clinical space. I'm sure, and you heard a lot if you were in the morning session with Mike talking about NAMs, and that's a big area, and we can really see where AI could support the development of NAMs, use in PK/PD, toxicology studies, as well as dose range finding. And then in terms of clinical research, and this is really probably one of the biggest areas where we're seeing the use of AI in terms of dose range finding, site selection, recruitment and retention of patients and trials, adherence, data collection, management analysis, real-world data analysis, and a lot of work happening in the clinical endpoints, talking about novel clinical outcome assessments, novel biomarkers, multimodal composite endpoints. And then in the manufacturing, post-market safety monitoring, a lot happening with AI in terms of advanced manufacturing, as well as for post-market surveillance and pharmacovigilance. So our core principles when we're talking about the use of AI in regulatory decision-making is that we want to make sure that we're being adaptive and that we're being technology enabling, that we're responsive, iterative to accelerate adoption and maintain patient safety. Of course, we know AI is ever adapting and changing rapidly over time, and we want to be able to be part of that and not sort of be behind the curve on some of those things. And then as far as the second principle is really a risk-based regulation approach. So not a one-size-fits-all regulation of emerging technology, but really having a data-driven, segmented approach that's really commensurate with the AI model risk. And then we're also really trying to be collaborative. So I kind of live that, living in both CDRH and CDER, and so we're collaborating across the agency on AI. And then, of course, we are also aligning across the ecosystem with patients, providers, biotech, pharma, other foreign regulators. I'll talk a little bit about that later. So how are we moving forward in terms of thinking about AI in the drug development space? So we're trying to foster, and this comes from the joint white paper we put out between CDRH, CDER, and CBER about how we can foster collaboration, that we want to advance regulatory approaches that support innovation, promote harmonized guidance, glossaries, guiding principles, and support research that is related to the evaluation and monitoring of AI. So this approach is informed by our expertise. So since 2016, between CDER and CBER, there have been over 1,000 submissions that have had AI in them. And this is where sponsors have flagged that AI has been used in the submission. And so we're seeing oncology is a big area on both the CDER and CBER side, with other areas certainly covered as well. And some of the example use cases are outcome predictions, confounding adjustment, external controls, post-market monitoring, dosage optimization, personalization, and post-market surveillance. And not only are we informed by our internal work, we're also informed by a collaborative engagement across the ecosystem. So in 2019, CDER launched the AI Steering Committee and Community Practice, as well as an AI Policy Working Group, as well as starting to track those use cases of AI. There have been multiple workshops, including a Duke Margolis expert workshop, a landscape analysis paper that is the basis of some of that initial data that I just showed you, discussion papers that we put out across on terms of thinking about how to leverage AI in drug development, additional workshops, the white paper I mentioned, that's the cross-center one, launching the CDER AI Council, and then the AI draft guidance, which I'll focus a lot of this discussion on. So this AI guidance is really the first one globally in terms of thinking about AI in the use in drug and biological products. So what the guidance provides is a risk-based framework to establish the credibility of an AI model for a particular context of use. And so when we think about credibility, we're really talking about trust that's established through the collection of evidence in the performance of an AI model for a particular context of use. And that context of use is the specific role and scope of the model to address the question of interest. So why did we publish this AI guidance? Well, from the FDA side, we were really trying to figure out, like, how do we determine if the AI risk is appropriate, if the AI use is appropriate for whatever specific context of use, and how do we determine the model risk? What do we think about in terms of evaluating the outputs, and what do we need to determine and communicate what we would need from industry in a submission? And from industry, what we really heard was understanding what's in and out of scope of FDA's oversight. How will we know if FDA will accept the use of that AI model? How much information or data will FDA be asking for, and how to engage with the agency on these issues? So the guidance table of contents, it goes through the intro. I'll kind of walk through these each in depth, but the focus is really about the considerations for the AI use in the drug product lifecycle. So we start by introducing some of the general terminology to sort of frame the rest of the guidance, and that ultimately the purpose of this guidance is to provide a risk-based credibility assessment framework to assess the credibility of the AI model for the particular context of use. The scope is to sort of really clarify, again, what we've heard from industry is, where is FDA going to look at the use of AI versus where is FDA not? And so what's in scope is AI that's being used to produce information that supports regulatory decision-making for the safety, effectiveness, or quality of the data. So what's out of scope is things like drug discovery generally, or operational efficiencies. So the easiest way I like to think about this is that process or activity that you did without AI something that FDA would look at? And if the answer is no, FDA is not going to start looking at it now just because you used AI. So that's sort of kind of the simplest way to sort of think about what's in and out of scope. And so this framework is really intended to help industry gather, plan, and organize the documentation to be able to support the credibility of their AI, the use of the AI model for whatever specific context of use. In terms of the background, we provide some examples of AI being used to support regulatory decision-making, kind of along the lines of what I talked about in some of the drug life cycle. So reducing, animal testing, integrating multimodal data, real-world data analysis. And we just talk through some of the unique challenges that are inherent to AI, like availability of fit-for-purpose data, interpretability, explainability, data and model drift, et cetera. So, and then the real meat of the guidance is really the considerations is the risk-based credibility assessment framework. And this framework is 7 steps. It's high-level concepts and principles that are based on FDA recognized consensus standards for computational modeling that we've been doing for a very long time on the device side. So it's not, the basis is really more on computational modeling, verification, and validation, which may not have been specific to AI, but then is tailored more specifically to AI. And the technical aspects of this document are consistent with the CDRH draft guidance that came out on the same day on AI-enabled medical devices. So these are the 7 steps in the credibility assessment framework. You have to start with defining the question of interest, then defining what's that context of use of the AI model, assessing the model risk, then developing the plan for how you're going to establish the model credibility, and most importantly, come talk to us so that we can discuss what might be needed in terms of ensuring that what information that the model is, commensurate with the risk. Execute the plan after you talk. Hopefully, you've talked to us before you did that. Document the results, and then determine the adequacy of the AI model for that specific context of use. And the guidance really focuses on the first 4 steps. So here's an example that's in the guidance that kind of explains sort of the model risk assessment and the credibility assessment framework. So drug A is under development and is associated with a life-threatening drug-related adverse event. In the previous trials, all patients went through a 24-hour inpatient monitoring after dosing. Data from the previous trial showed that some patients were at low risk for this severe adverse event. And so in the new study, the sponsor now wants to use an AI model to identify low-risk patients that could be monitored at home instead of at an inpatient facility. And then as we talk through this example, one key feature is that they would be relying solely on the AI model and not necessarily having a clinician in the loop. So the first question is in terms of you're defining the question of interest. So in this case, it is which participants can be considered low risk and don't need inpatient monitoring after dosing. So then based on that, we think about what's the role of the AI and the scope of the AI. So the role of the AI here is to stratify patients into low versus high risk. And then the scope is that only the model is being used to determine the monitoring type. Then we'll talk about assessing the model risk next. And so the credibility assessment framework is a 2x2 basically of decision consequence and model influence. And so this model risk matrix transitions from low to high as decision consequence or model influence increase. And these 2 factors are determined independently. And so in this example case, the model risk is high because both the decision consequence and the model influence are high. So the reason the decision consequence is high is because if you mischaracterize a patient as low risk, but they actually are high risk, that obviously could result in a severe adverse event. And the model influence is high in this case because the AI model is the only determinant of which monitoring a patient gets. So a way you may lower the risk of this AI model is if you are including a clinician in the loop and a clinician is making the final decision on what category to stratify a patient to, low or high. So that may, change the risk profile in terms of the model influence. So when we think about activities to establish the AI model input and how it's commensurate with the model risk and tailored to the context of use, we are thinking about things, like I said, level of oversight. So, is there a clinician or another human in the loop? How stringent the credibility assessment and performance acceptance criteria are, what other risk mitigation strategies might be associated with the model, and then the type and extent of documentation and detail associated with the AI. Then you're going to develop your plan and come talk to us. And when you're thinking about that plan, when and if and how you plan to submit really depends on how you engage with the agency and on the context of use. So the plan could be in a formal meeting package, or it might be in another type of an engagement opportunity with the agency. So we hope to envision that it's interactive feedback, even if you don't have all the details of information, but at least at a minimum that you have steps 1, 2, and 3 articulated. So that way we can give you some feedback. If you just come in and say, we want to use an AI model, we're going to be like, okay, cool. I don't know what else to tell you with that level of detail. So, the more you can give us, the more we can respond. And then, what we ask for in step 4, again, is consistent and aligned with CDRH efforts. And so we're able to learn across centers. In terms of what we ask for in step 4 is to really describe the model and the model development process. So thinking about what are the model inputs, outputs, the architecture, features, rationale, et cetera. What was the developmental data in terms of what training, tuning that was used for the model? How was the data collected and processed? Is it fit for that context of use? How was the model trained? Any performance metrics? Is it using a pre-trained model? Describing any calibrations. And then as far as the evaluation, describing that as well. So how did you as a sponsor describe the evaluation of the fully trained model? What test data was used? Is it independent from the training data? Was it applicable to the context of use? Any other performance metrics or estimates? Then, as I said, really steps 5, 6, and 7 are for a sponsor to execute on. And then the next section of the guidance, we talk about some of the special considerations of life cycle maintenance in terms of the credibility. And so that's going to be more important in certain stages of the drug product life cycle and management of changes to the AI model to make sure it still remains fit for use in whatever context of use, and that we're taking a risk-based approach for lifecycle maintenance that can help sponsors assess any impact of any change or changes in model performance. And then we talk about early engagement, and really, again, I don't think I'm going to sound like a broken record here. We encourage early engagement with FDA to set the appropriate expectations, to think through what is really going to be appropriate for the credibility assessment activities. And there are a variety of different pathways that you can use to engage CDER, which is always interesting to me because my CDRH brain is like, we just have pre-subs. But in terms of CDER, there's various opportunities depending on what type of context of use you've got and you're planning to use your AI model. So for certain non-clinical or even clinical research, the model-informed drug development program might be the right pathway depending on how you're using the AI model. In terms of in clinical research, it may be related to real-world evidence, digital health technologies. We know there's a lot happening in terms of using wearables and other sensor-based technologies in clinical trials, and usually there's AI involved because otherwise the raw data from, accelerometry on its own isn't really going to be helpful. So you need to have the AI algorithms on top of that. There's also the complex innovative trial design meeting if you're talking about more in the clinical trial design space and using AI models. And then of course there's C3TI, the Center for Clinical Trial Innovation, where there's opportunities for demonstration projects, and AI is an area that people can propose demonstration projects. In terms of manufacturing and post-market safety opportunities, and you'll hear much more about the Emerging Drug Safety and Technology Program from WANS, and then CDER's Emerging Technology Program, and then CBER also has the Advanced Technologies team. In terms of additional work that we've done in terms of thinking about how we're sort of fostering collaboration, we have also worked collaboratively with the EMA on guiding principles for the use of AI use in drug and biological product development, because we see, as I've talked about already, the increased use across the entire life cycle. And for AI to fully realize its potential, reliability, transparency, and patient safety have to be assured, and that these principles really provide a common foundation for good practice, international collaborations, and future standards. And again, similarly to the AI draft guidance, this builds on good machine learning practice that CDRH has originally put out with MHRA and Health Canada that then became adopted by the International Medical Device Regulators Forum, or IMDRF. So these 10 principles kind of really do cover across the entire life cycle and are kind of focused in 3 big bucket areas. So design and governance, data models and expertise, and assessment and oversight and transparency. So when we're thinking about design and governance of AI, you want it to be, of course, human-centric by design, and that we really are reaffirming across the globe that it really is a risk-based approach to the use of the AI. Adherence to good standards is a way to ensure models are credible and are fit for purpose. And then, of course, ensuring you have a clear context of use. In terms of the data models and expertise, one of the principles is about multidisciplinary expertise. And this is a common theme across the board of you want to ensure that you've got not just your data science AI experts, but you want to have them with your clinical, your pharmacology, your other stakeholders who are involved in whatever that context of use is, so that you've got the right expertise. So you're not just building a model for the sake of building a model or using a model for the sake of using a model, but it's really something that is fit for purpose and is going to help with your drug development program. ensuring you have appropriate data governance and documentation, and then of course, following good practices for model design and development. And then in terms of assessment, oversight, and transparency, again, really focusing on that risk-based performance assessment framework and thinking through it from a life cycle management perspective, not just at one point in the use of the AI, and that you're also ensuring clear and essential information is transparent to the user of that AI model or how it's being used in the drug development process. So in terms of what's next, as we continue to collaborate, as I said, across the agency, we are collaborating between the different medical product centers on discussion papers, workshops. We're working to finalize the guidance based on a lot of feedback we got. We are continuing to work across other regulators. So we have regular bilateral engagements with EMA to share mutual learnings. We co-hosted a workshop a few years ago with the Korean FDA, the Ministry of Food and Drug Safety, on the AI Regulatory and International Symposium for using AI in medical product development. We're continuing to promote harmonized glossaries, guiding principles, and standards. And so, again, we're collaborating across the agency. We put out a glossary a couple years ago, the FDA DHT and AI glossary, and that will continue to be updated at various points over time. And then we're also engaging bilaterally with EMA on taking some of what's in the glossary the FDA posted and seeing where it can be mapped to EMA. And then in terms of advancing regulatory approaches that support innovation, as I said, we are working to finalize the guidance. I'm sure many of you in the room provided comments. We got a lot, over 1,400 from 98 different entities, and went through them all, and we're working to address them and get the guidance finalized. And hopefully, I'm not going to prognosticate a timeline because that's never a good idea with clearance. So with that, I may have ended early, but I can take questions.
Thank you very much, Annie. Okay, any in-house questions? Did I hear someone say yes? Please step up to the mic, please. Just that.
A couple of years ago in one of your conferences, a radiology engineer gave a talk, and someone in the audience asked him to please define synthetic data. Is there any further discussion about synthetic data at FDA?
Yeah, so I think that's an area we see there's a lot of potential for sort of streamlining clinical trial design, both in the pre-market and even both preclinical and clinical. And there has been research that's been done across the agency, both in CDER and CDRH, about exploring the uses of synthetic data. And you could certainly see, especially in like rare disease context of being able to supplement with synthetic data could really be helpful to get those types of products to market.
But synthetic data comes from real data, right?
It can. It can also be done in sort of a research environment. So, for example, our Office of Science and Engineering Laboratories on the device side has a laboratory where they have like lung nodule, like a 3D printed lung and different sort of nodules. And so you can create sort of synthetic test data sets using, for like lung nodules as an example.
Okay, and I just have another general question with regard to integrative medicine. Does anyone, you guys have given a couple of approvals lately based on the drug plus nutrition. Is there a critical mass of companies that control their results for the nutrition as well?
I am not an expert in that area to be able to say where the trends are on that.
Okay, thank you.
Thank you. Question in the house, please.
Yeah, thank you. What is the correlation between this work and ELSA utilization by the FDA?
So this is really, this guidance and a lot of the work that we're doing in the Office of Medical Policy and CDER is really focused on how we're thinking about AI that's being used in products that FDA regulates. So it's about the policies for how we look at AI when it's being used in as part of, say, the regulatory decision making for a drug. Ultimately, and this is where it's this difference between drugs and devices is, ultimately the regulated product at the end of the day is the medicine itself, but you may be using AI to support whether it's the clinical trial design, et cetera. And so we'd be looking at how AI is used as part of that process, but that's not the same as the internal use of tools that like ELSA. So ELSA is an internal tool based on Claude. It's in a FedRAMP high environment, and it's a tool to assist agency staff to help, with sort of improving operational efficiencies.
Thank you. Do we have any online questions?
We do. So the first question states, Annie, can you speak to the recent warning letter that included a violation based on the use of AI and the failure to verify the output?
Yeah, that's an interesting one. The LinkedIn takes I saw right when it came out were like, FDA is regulating AI and inspections. And it's like, okay, let's take a step back here for a second. Let's say you hired a bad consultant who didn't tell you to do some part of the GMP. And FDA comes in, the inspector comes in and asks why you didn't comply with that part. And the response of the company is, well, our consultant didn't tell us that. The liability is still on the company to follow all the manufacturing processes. So it's the exact same. So the response would have been the same if you said you used a bad consultant versus you said you used AI. Ultimately, at the end of the day, the responsibility is on the company to follow all the manufacturing requirements. So it's not about the use of AI, it's about not following the manufacturing requirements.
Thank you, Annie. Next in-house question, please.
Thanks, Andy, for the presentation. Really great overview. And I asked this question to Anish, but he kindly referred me to you as well, because it's about use of AI across entire, drugs and medical devices, agnostic. So I know, being from the DHCOE and now in OMP, you wear these dual hats that are quite advantageous to your overview of how the agency collaborates. Do you think that there's going to be more opportunities to facilitate these early engagements that you stress so often that companies and sponsors do when thinking about these innovative uses of AI that are often paired with, DHTs that may be used in clinical trials and even beyond clinical trials as SAMDs themselves? How can we bring kind of like these innovative approaches from both agencies? Because I think the pre-subs are very actually useful for conversations directly with reviewers. Although CDER has a lot of engagement opportunities, very few are directly with the reviewers themselves to discuss, opportunities, unless you have like a submission already ready. So just a couple thoughts, if you wouldn't mind.
Yeah, and I think with that, there's always opportunity as you're coming in with whichever engagement pathway to ask for additional folks. So if you're coming into CDER about the use of AI and you think it will be helpful to have like CDRH there, you can ask for that in your cover letter. And so that may be, or if you want like certain review teams there, you can ask for all of those aspects in your, in your request to meet with the agency. And then, vice versa, if you're coming into CDRH for, a medical device that you're developing that you think may have implications or use, like say in a drug clinical trial because it's a wearable that could maybe be used in trial, you can ask to include, the CDER review team as well. And so I think that's a good way to be able to ensure that you've got the right folks from the agency.
Thank you, Annie, for that response. Any in-house questions, please?
Yeah, thank you so much for this. This guidance is so helpful. I'm wondering a little bit more about how this Claude-based ELSA, is it switching over to Gemini then in this current context? And how does this guidance increase operational efficiency and like the ways that we still interact with FDA then?
It doesn't really change. I mean, the use of ELSA doesn't change how we are going to interact with sponsors on sponsors' use of AI in their regulatory decision making.
Oh, I guess, sorry, that's actually really useful. I guess what I'm asking is, so we have a product that's not AI based. This is just a standard drug development program. So would ELSA still be involved on your end? Is there still this AI involvement in the review process, even, independent of, the product being AI?
Well,
Or not?
AI is ELSA is a tool that can help, and people do use it. Not everyone uses it, and not everyone uses it for the same use cases. But ultimately, at the end of the day, it's the review team is making any decisions. So if they want to use AI in some way to support, it does help with administrative tasks of trying to find, something on page 8,478 of your document, and you're looking for, in the submission. It's very helpful to get you to what you need and what you need to find. So, there's a lot of operational efficiencies.
Yeah, thank you so much. It's really useful to get that example.
Thank you. Any online questions there?
Sure. Our next question states, there appear to be more FDA guidances on AI/ML from CDRH for medical devices than with CDER. Is the assumption that these AI/ML guidances also reflect FDA's current thinking in CDER for drug development and clinical trials?
So yes, there's a lot more guidances on the CDRH side in terms of AI and digital health, because they are, in this case, regulated products that we are actually reviewing and not making a marketing decision on. And so there's a lot more to be thinking about when you're talking about a product going to market versus on the drug side. And why it's really only one guidance about the AI use in drug development is because ultimately, at the end of the day, as I said before, the regulated product is the medicine, not the use of the AI. And so that is why there's a lot more guidances on the CDRH side. And again, that's where the sort of coordination and collaboration between the two centers splits in. And so the CDER AI guidance is based and has had conversation, and CDER and CBER and CDRH have all had conversations about how to leverage a lot of the learnings that we've had on the device side for how to assess AI in drug development, because if you've seen our list of AI-enabled devices, it goes back to 1995. So CDRH has a lot of historical experience with AI, and so that's being leveraged in terms of how to support the other centers.
Thank you for that. Any in-house questions? We do have some minutes in our presentation time. Do you have any online questions there for us, Corey?
We have another online question. Says, what extent of documentation is required in this credibility assessment framework?
Everyone's favorite answer, it depends. So it's going to depend on the context of use, so what you're using the model for, and then the level of risk. One thing to maybe also touch upon on level of risk, like just because a model is high risk doesn't mean you shouldn't use it. You just have to provide the appropriate documentation for the use of that AI model. In fact, it may be a game changer. It may be really helpful for you to use a higher risk model to really support your drug development program.
Okay, we have about 5-7 minutes. Let's go ahead with any online questions that we have right through. Unless someone in the room has a question, please step up to the mic so I can see that you have a question. Corey, please.
Sure. Our next question is, are there further plans to collaborate with other international partners?
Yeah, so we are exploring, what does that look like? Obviously the bilateral engagement with EMA is fantastic, but I think there's opportunities, whether it's through ICH, ICMA, et cetera, what does that look like? And, where can we learn from, where can regulators kind of learn from each other, but then also where can we learn from, what sponsors are doing in terms of using AI in drug development. And one thing I will also say is part of, slight aside, but kind of related is, we do want to hear from you as you all are using AI and getting use cases out and more examples, not just from the FDA side is that we put in guidance, but the more that we're hearing and seeing and the more that you're willing to share, I think can help the entire industry move forward.
A moment. We might have one for online here. Let me see. Says, Where can we learn more about digital twins and how they are used? Have you, from FDA's perspective, evaluated such scenarios? And can we see or hear how you walk through the review of those scenarios from the agency's perspective?
I mean, as far as how we would review, digital twins, it would be similar to how we'd review any part of a drug, submission in terms of how it's being used in relation to whatever specific context of use in the drug development program. And as far as that is a use case we have been seeing as well in terms of that over 1,000 submissions of AI being used in drug development, digital twins, synthetic data. Those are aspects that we have been seeing. And then, again, we review it as we would any other type of part of the application for, ensuring you have valid documentation, you're following good science, and then how it's being used sort of in the context of the assessing either the safety, effectiveness, or quality of the drug.
Thank you. Is that it for online?
At this time, unless more come in.
Okay.
For this.
Is that it for in-house? I see people vigorously nodding their heads. Yeah. So do you have a question or are you stretching your legs there? Okay, question.
I have a proposition. This
You have a proposition, you said?
AI helps more FDA or the industry?
I'm sorry.
AI helps more FDA or it helps more the industry?
I think AI can certainly help FDA. It is, with some of our operational efficiencies, but I think it's.
I think AI helps FDA more than the industry because industry cannot move alone. Industry has to be under your purview, but you can make anything that works for you using AI. Is there any legal binding for that?
Sorry, I think I didn't quite understand the question.
I mean, how much AI will be used by FDA to review? Does it have any guidance, any limit of that?
So we do have AI governance and best practices for our staff.
Governance on FDA side or on industry side?
Well, it's not our authority to.
No, is there any guideline to limit both sides?
I'm not.
That would be a work in progress as it advances, as AI progresses and technology progresses. We have to see where it leads us, right?
Yeah.
Yeah. I mean, ultimately, at the end.
It seems AI helps FDA more. I mean,
and that is one perspective.
If some company uses AI, then they cannot do that without permission of FDA.
Okay, so this is a forum for questions, not for, hypothetical discussions. So thank you very much for giving us food for thought. Any questions online?
We do have one question online that asks, does FDA promote any specific technology approach?
No.
We love
okay.
That's an easy one.
All right. After that, we'll say thank you very much, Annie. We welcome Dr. Dang, Dr. Wan Dang to the I was going to say pulpit, but to the podium to speak with us about FDA CDER Emerging Drug Safety Technology Program. Thank you, Dr. Dan.
Okay, thank you, Brenda, for the introduction. Good afternoon, everyone. Today, I'll be going over what we learned from the first year of the Emerging Drug Safety Technology Program, specifically the meetings that we had with external stakeholders. And with these meetings, there is a particular focus on the use of AI for PV. And these meetings are a part of the Emerging Drug Safety Technology Program, which is only one of CDER's multifaceted approaches to facilitate mutual learning on where and how innovations such as AI can be optimally applied across the drug product lifecycle. Just a disclaimer slide. So I'll first start off by providing some background on pharmacovigilance and what is driving the use of AI in PV that we're seeing across industry. I'll then go over what led to CDER establishing the EDSTP program, as well as its purpose and goals. Then I'll discuss what we learned and observed in a 1 year. You'll learn about the participants who were involved, the types of use cases, and AI technologies being applied to PV. Also, I'll provide some observations of approaches related to AI model credibility assessments. So just for background, once a drug is approved and marketed, FDA ensures that the safety of drugs through ongoing surveillance. So in the post-market setting, regulated industry is obligated to review all adverse events, adverse drug experience information received, otherwise obtain and submit in individual case safety reports or ICSRs to FDA. So this brings us to pharmacovigilance or PV. Like Annie said, it's the whole drug product life cycle. And PV describes all the activities, all the scientific and data gathering activities related to detecting, assessing, and understanding of adverse events. These reports or individual case safety reports are called ICSRs, and they're reports that really capture details about the adverse drug experiences by patients, which have both structured and unstructured fields. And these ICSRs are an important data source for ongoing post-market drug safety surveillance because they enable the detection and evaluation of adverse event safety signals. And especially, they allow us to see what is really happening in the real world setting where there's more widespread use of the drug and in broader patient populations. So as you can see here to the far right, as of mid-May of this year, the FDA Adverse Event Monitoring System, also known as AIMS, has over 3 million ICSRs. And in recent years, we received over 2 million reports annually. So this is quite a large volume. Now, from industry's perspective, the process of case intake and case processing, evaluation, and submission of ICSRs to FDA is time and labor intensive. This is a multi-step process. Some of the steps could include data extraction, language translation, medical evaluation, and even ICSR narrative writing. So it's not really surprising that this process requires significant human resources, technology, and budget to meet regulatory reporting requirements. So why is there a need to apply AI and emerging technologies such as AI to PV? So as mentioned, there are challenges of large and increasing ICSR volume, and the data is coming from a wide variety of sources. In parallel, there are emerging technologies such as foundation large language models or LLMs and agentic AI that could bring new capabilities to the PV workflow. So for instance, with LLM, they can process, analyze, and generate large amounts of complex safety data. And AI agents have a certain level of autonomy to support multi-step PV processes. From an industry's perspective, what we're hearing is that there are questions about how do we effectively use AI for PV, what will regulators accept, where to optimally invest resources. And from a regulator's perspective, FDA expects that increased communication with the broader pharmaceutical industry during EDSTMs will accelerate FDA's understanding of how AI-enabled tools and other emerging technologies are being used for PV. We will be able to understand their associated risks and benefits, model evaluation processes, including performance characteristics, and even barriers to implementation. So to do this, we recognize that it is important to have visibility on how AI is actually being applied to PV in the real world, which means engaging directly with and having discussions with external stakeholders. So this led to the establishment of CDER's Emerging Drug Safety Technology Program, or EDSTP, in June of 2024. Where the purpose was to proactively understand how emerging technologies like AI are being used in PV and position the agency to respond as emerging technologies develop. So there are 3 goals. one of which is to facilitate dialogue, serve as a central point of contact between industry and CDER on emerging technology in PV through the EDSTM engagement pathway. 2, to build knowledge, enabling knowledge management and transfer within FDA, as well as to inform potential regulatory and policy approaches as emerging technologies within PV. Just want to note that the EDSTM program does not cover the application of emerging technologies outside of PV, and that the program is not an avenue to seek regulatory advice regarding the adequacy or acceptability of drug technologies that uses AI or other emerging technologies. But rather, we expect that the information gained from the program will help CDER consider providing regulatory advice on specific technologies to facilitate their adoption when appropriate. And I just want to mention that FDA will not proactively disclose details from these EDSTMs meetings, including participant names. However, the agency may share aggregate information such as the number of patients and general topics covered. So the discussion and background information submitted through EDSTMs are also non-binding on both FDA and EDSTM requesters. So the Emerging Drug Safety Technology Program administers these meetings, and EDSTMs can be requested by applicants with at least one approved application regulated by CDER, including new applications like NDAs, abbreviated new drug applications, or biologics license applications. And also the eligibility is also extends to other relevant parties supporting industry's PV activities, including academia, CROs, pharmacovigilance vendors, software developers who develop and leverage or intend to leverage AI or other emerging technologies that can be used to satisfy the post-marketing reporting requirements. So eligible applicants and other relevant parties can request meetings, and the aim really is to have a balanced portfolio of AI use cases for BV that would really enable us to get a better picture of how people are using AI for PV in this space. So we're looking to engage with organizations that had AI use cases at different stages of development, including research, pilot, development, and even production. We're also looking for a variety of use cases across the PV workflow and to understand its implications for regulatory obligations. We also want to understand different technologies and their approaches, as well as their potential broad applicability. And what we really want to see, too, is how organizations are approaching establishing AI model credibility. So during our first year of EDSTMs, we received 20 requests for EDSTMs, and that to us demonstrated a high level of interest in this area. And then we held 9 meetings. The requests came from EDSTMs. The requests came from a variety of organizations, including vendors and companies that develop AI tools. We also heard from pharmaceutical applicants, the companies who are responsible for pharmacovigilance activities, including meeting reporting requirements. And finally, we also heard from academia, who are researchers who are exploring novel technologies and approaches. So EDSTMs enabled us to see a variety of use cases across different parts of the PV workflow, with the most common AI use cases being intake, which included data extraction from a variety of sources. Another area was case processing, in which AI was used for coding adverse events, as well as the use of AI for generating ICSRs. We also saw safety profile monitoring was another area for signal detection. And finally, we also saw AI being applied to support causality assessment between a drug and an adverse event. So EDSTMs also enabled us to see and understand the different AI technologies and approaches that are used in PV. We saw a wide variety of AI technologies being employed, where the most common was large language models, then this isn't really surprising considering the LLM's capabilities. There were also smaller, more specialized models like BERT-based models that were fine-tuned for specific domain tasks, all the way to larger commercially available foundation AI models. So the LLMs were used in different ways. Some organizations use LLMs in a non-generative way. So for example, using LLMs to identify and extract specific data elements from unstructured texts, such as reporter information, adverse event, or drug name. We also saw LLMs that were used generatively to create new content, such as generating case summaries. We also saw hybrid systems where LLMs were paired with a role-based approach to leverage really the different strengths of the different technologies. And we saw LLMs being deployed as agents where there was a coordinator agent that would orchestrate multiple specialized agents for specific tasks. And then other approaches were traditional machine learning, such as classification tasks, as well as ensemble methods that combine multiple models. So what's important to keep in mind here is that different AI approaches can be leveraged for PV, but companies remain responsible for ensuring that their approach is appropriate for the specific use case. So we engage with organizations through the EDSTMs, where the AI use cases were at different stages of development, including research, pilot, development, and in production. The majority of the use cases were in production or active use, and some were in the other stages. And then we even had some organizations who had multiple use cases at different stages. So this slide right here highlights the key PV challenges that were discussed during EDSTMs. The first challenge area was discussed was the significant manual effort required to process safety data. We know that adverse event information does come from different sources and is often unstructured and requiring extensive manual extraction and review. At the same time, adverse event case volumes continue to grow, which then creates challenges for case intake, case processing, evaluation, and reporting activities. The second challenge area that we saw was the medical evaluation of adverse events. That means the assessment and interpretation of adverse event drug experiences often involve complex clinical and patient scenarios that really require medical expertise. Additionally, causality assessment can be complex and resource intensive because we know PV experts need to apply clinical expertise and judgment to evaluate whether the reported adverse event is associated with the drug or is it associated with the underlying condition or another concomitant medication. So it's really this contextual dependencies that make these analyses challenging even for human experts. So overall, these challenges contribute to the increased interest that we're seeing in the using AI for PV. So this slide highlights examples of how AI might be used to help address some of the PV challenges. AI-enabled multimodal processing may help automate extraction of safety data from multiple heterogeneous and unstructured sources, as well as support rapid and scalable processing of growing volumes of adverse events and streamlining case intake and processing activities. AI may also be used to support quality improvements by supporting human experts' assessments of the adverse drug experiences and enhancing the consistency of medical evaluations, such as using AI to apply more standardized criteria across cases to help reduce variability in the medical evaluation process. Although AI may help to improve the quality and the completeness of ICSRs through better data extraction and identification of missing or inconsistent information. So as discussed by Annie, the previous presenter, in January 2025, FDA published the draft guidance to support AI use in the drug product lifecycle. And this is applicable to PV. And so several concepts discussed during EDSTMs align with the guidance. Risk-based credibility assessment. This guidance provides recommendation in industry, on the use of AI to produce information or data intended to support regulatory decision-making, which includes safety, efficacy, and quality for drugs. So for reference, this is just a visualization of the 7 steps of the risk-based credibility assessment framework from the guidance that Annie had well articulated earlier in her presentation. So I won't go through that. And so building on FDA's AI draft guidance, the next several slides provide some observations of examples of AI model credibility assessments that were discussed during the first years of the EDSTMs. So in particular, these observations align with concepts from the guidance related to assessing model risk and approaches for establishing AI model credibility activities based on the context of use and model risk. So in the following slides, I'm going to provide a high-level overview of highlights and approaches and considerations that we observed across different PV AI use cases. So when we think about assessing AI model risk, an important consideration is where and how AI's output is used within the PV workflow, and whether there will be a human in the loop. In other words, what is the role and scope of AI, which is essentially the context of use. A second consideration is model influence, which is to what degree is the AI output used to inform regulatory decision-making. And this has to be taken relative to other contributing evidence or with whether there's a human review or not. So for example, a PV example is if the AI is used to help prioritize cases for human review, the model influence is generally lower if the AI the model influence is lower if humans are also involved in the review. And third, we need to consider the potential decision consequences if the AI output or AI-informed decision is incorrect. So what that would mean in terms of the potential impact on patient safety. So in PV, both false negatives and false positives have important consequences. False negatives, where the AI fails to identify a true safety signal, could lead to safety concern that might not even get investigated, which could then put patients at risk and worse, put patients at harm. False positives, where the AI incorrectly classifies a safety issue, could increase unnecessary workload for PV experts who then have to reconcile the AI's inaccurate output. So this also can delay the identification of safety signals. So overall, the model risk is commensurate to model influence and decision consequence within the context of use. So AI model performance significantly relies on the data sets that are used for model development and evaluation, right? You're thinking training, tuning, testing, and evaluation overall. So therefore, these data sets that are to be used should be fit for use. And some of the considerations discussed at EDSTMs for establishing AI model credibility include the use of fit for use data sets for AI model development and evaluation in PV workflows. These data sets should be relevant for the intended context of use, representative of the real world PV data and workflows, and be reliable in that they're complete, accurate, and traceable, all of which directly impacts AI model performance, reliability, and credibility in PV workflows. So when establishing AI model credibility in PV, it's important to recognize that AI model performance may change over time as PV safety data and reporting patterns evolve. We know there's new emerging adverse events sometimes, there's new drugs that are coming into the market. There's also changes in the coding of the medical terminology. So all of that needs to be taken into consideration when we talk about PV. And this is especially relevant for adaptive models in PV, which may need to be periodically retrained using new safety data or literature, and even using reviewer feedback to help maintain the performance of the AI system within this dynamic PV environment. Now, because these models can evolve or adapt over time, proactive monitoring and lifecycle maintenance are important to help ensure model performance and credibility is maintained within expected outcomes within the intended consequent or context of use. We also observed PV AI use cases where performance evaluation was conducted at the level of the individual models or individual agents. However, for complex AI systems involving multiple models or multi-agent workflows, the evaluation may extend beyond the individual component performance to also how multiple AI models, agents, tools, or systems interact with each other and how that information is handed off between the components. And then over the overall, and also thinking about the overall end-to-end performance of the AI system itself. So I just want to mention that an important consideration here is that even when individual models perform well independently, issues or errors may still arise from the models interacting with a larger integrated AI system. AI performance is another component of establishing AI model credibility. Performance can include quantitative or qualitative performance measures, as well as determining what level of performance is acceptable. What's important to note here is that there's not a single performance metric or threshold that applies across all AI use cases. Acceptable performance threshold really depends on the context of use and the level of model risk. But generally, as the AI model risk increases, the expectations for AI model credibility, including acceptable model performance, also increases. Other considerations include when the context of use, if that involves a human in the loop, evaluation methods should also consider the performance of the combined human and AI team. Finally, the quality checks and oversight may help to ensure that the combined human AI system perform at least as well as the human-only process it aims to replace. So another consideration is establishing AI model credibility in PV relates to human oversight. When evaluating AI outputs in PV, human oversight may involve applying scientific expertise and clinical judgment to assess the accuracy, quality, and appropriateness of AI outputs within the intended context of use. The level of human oversight depends on the context of use, and in general, higher model risk use cases may require more human oversight. Finally, we observed approaches where ongoing human feedback was incorporated to support model refinement and improvement over time. We also observed user-centric design, interactive user interfaces that was used to support human review and usability in PV workflows. The UIs may enable human experts to review, correct, modify, and verify AI outputs for accuracy, as well as to also be able to compare AI outputs with the source data. While also at the same time, the user interface was able to capture the ongoing human feedback for system improvement or AI model refinement. Overall, a thoughtful user-centric design user interface has the potential to improve efficiency and effectiveness of human review and verification of AI outputs. So evaluation doesn't end once an AI system is deployed. Ongoing lifecycle maintenance is important to ensure that the AI system continues to meet expected outcomes such as performance and regulatory requirements. And as mentioned in PV, due to the nature of evolving safety data, AI performance may change over time. So therefore, it's important that there's lifecycle maintenance of these AI systems, and during the EDSDMs, some discussions included evaluations that define time intervals, and that the frequency of the evaluation was tailored to the context of use and the model risk. In general, for high model risk, there's generally a shorter time in between the evaluations. Another aspect of lifecycle maintenance of AI model credibility is periodic model retraining or refinement. Here, we observe the use of human user feedback and new safety data that's used to either maintain or improve the AI performance over time. So for example, increasing error rates could trigger periodic model retraining or refinement. And finally, we observe the importance of documentation to help understand the monitoring processes and activities, what issues and errors were identified, the number and nature of disagreements between the human reviewer and AI output, as well as the process for capturing human feedback. And also, the documentation helps with understanding the analysis that were involved and the subsequent resolutions that followed, including how the disagreements were handled and the resulting actions that took place. And what was also helpful was to understand if, how, and why any changes to the model was made, and that would not only support continual AI improvement, but also enable traceability. So overall, from the first year of EDSTMs, we observed a wide variety of AI uses in PV across different stages of development, as well as varied approaches to applying risk-based credibility assessment framework. A key observation was while multiple AI approaches can be applied to PV, they should really be fit for purpose, as well as ensure timely, accurate, and complete information is submitted to FDA. We also observed that establishing AI model credibility needs fit for use data, human expertise and subject matter expert involvement, as well as considering that evaluations can occur at multiple levels of the AI system. And finally, ongoing lifecycle maintenance is important to ensure that deployed AI systems continue to meet expected outcomes and regulatory reporting requirements over time. So looking ahead for EDSTMs, we anticipate the use of AI and other emerging technologies in PV to continue to evolve. One of the key observations from the first year of EDSTMs is the value of discussions about AI and PV in a mutual learning forum. EDSTMs provided an opportunity for FDA and external stakeholders to exchange perspectives, discuss challenges, and share practical experiences related to AI and PV. We also anticipate continued stakeholder engagement moving forward, and there are opportunities for stakeholders, as you see here, to contribute to the broader understanding of AI applications in PV, and that will help inform potential regulatory thinking and policy approaches in this space. Overall, the goal is continued learning and dialogue as AI and other emerging technologies and their applications in PV continue to grow and mature. So I would highly encourage you all to consider submitting a request for an EDSTM, whether you're researching or developing or using emerging technologies in PV. There's a QR code on this slide here that it takes you directly to the webpage with more information about how to apply and other aspects, and even an email that you can reach us at. The next deadline is July 1st, 2026. So FDA in general will grant EDSTM requests quarterly each calendar year for a total of up to 9 participants in the 12-month period for this initial phase. And then there's a quarterly submission deadline, as you see here, and eligible applicants and other relevant parties may submit EDSTM requests on a rolling basis. So we'll review them in a 3-month submission cycle after each submission deadline. So I want to acknowledge my FDA colleagues for all their contributions and efforts with the Emerging Drug Safety Technology Program and meetings. Thank you.
Thank you very much, Dr.
I'm happy
Dang.
to take any questions.
Thank you very much, Dr. Dang. And let's open up the floor for questions, please. Let's start with in-house questions for those of us who are still here. How lovely, we still have quite a few people still here. Any in-house questions? Next year, I'll bring you a prize and, bribe you for questions. Okay, here we are.
I love prizes, so that works.
Okay.
So thank you for this presentation and also for sharing further opportunities to participate in the EDSTM. I do wonder though for US sponsors, obviously many of us will use vendors, and you described that there are many vendors participating in the EDSTM. And those vendors often outsource to other countries, and the PV process is often highly complex because of differences in both medical training and understanding of patient populations, even with those same diseases for comparison purposes in those other countries. It's very challenging. So is there an effort right now to rectify this in any way, or any encouragement of US-focused assessment in that regard?
Yeah, so let me just rephrase the question. What I'm hearing is that you're saying that for the most part, pharmaceutical companies do rely on other vendors for the pharmacovigilance activities because there are certain specific nuanced medical practice, for example, in different areas or even different medications that are being used in different countries. And so then the question becomes, you're asking has to do with whether there's any effort to make it more US focused. Is that how you, and more standardized?
It's really a big challenge for us to be able to produce data that's meaningful, especially in larger, like phase 3 trials or many others, and using even teams that are based in other countries. And so if we're looking to standardize the data outputs from this work, it would be important, I think, to review.
Yeah, thank you for the input on considering the standardization of data. I think how we're approaching this is we're relying on the AI guidance to be applicable for the PB activities, and you'd have to think really about the context of use as well as the model risk. And so I think the standardization part, if you will, is more of the application of the guidance, and then because each country or PV activity is very different, like you say, that will probably be the unifying way to apply the guidance for each of the PV activities that happen in different countries. Thank you.
Thank you very much, Juan. Any more in-house questions? Please don't wait for me to call you. Come up to the mic so I can see that you have interest in a question. In the meantime, any online questions, please?
Of course. Our first question is, what are expectations for explainability of AI systems?
Thanks. That's a good question. I think explainability has kind of been on our minds a bit, especially given that some models are, especially the complex ones, are quite difficult to explain, but also difficult, or even you can't really explain how they arrived at their conclusion or the AI output. So they're kind of like black boxes. And because of this, the guidance does emphasize the use of methodological transparency, which is how you would then be documenting the processes and the methods for how the model was developed, how it was evaluated, how it's deployed and maintained in that process. And that helps with the explainability aspect. And then in my view too, I think the UI, a very user-centric design approach that I described earlier, can also be used in PV to help support explainability from the end users. Because a lot of times if you have an AI system that then provides you with, say, it's going to be used to help prioritize ICSRs or cases for a human reviewer to look at, the end user generally would ask, well, how did it arrive there. So a UI would allow some visibility into thinking about what features were involved or what factors, and be able to sort of visualize that to help them make a more informed decision for their PV activities. There is one thing I do want to mention too. I think the first person who asked me the question, it reminds me that I think at the end of the day, it's the accountability. It lies in the pharmaceutical company for meeting timely, accurate, and complete reporting requirements.
Thank you very much. Let's go to online questions, please, unless we see someone step up to the mic.
Of course. Our next question is, what are effective strategies for AI integration into workflow?
Yeah, so you're asking about effective strategies for AI integration in workflow. I think that's really a really important point that can help to increase user adoption and trust and build trust among the end users. I think understanding how AI fits operationally within the real workflow and how humans interact with the UI is the AI output is really important. And so this process really would involve the end users in every step of the AI development and deployment lifecycle. Because what you need to understand through that interaction is to think about, or to understand rather, how their workflow works, what's their current workflow, and where to optimally insert that. And you're really only going to get that through conversations with them. In addition to that, you also have to think about making sure that you're using the AI to solve a problem that is meaningful to them, right? So you really want to solve a problem that they have, not the problem that you think they have. So that's, I think, really key. So really, by thinking about how to best integrate it into the workflow, you'd also need, in addition to SMEs, you would also need a multidisciplinary approach where the AI scientists, the SMEs, and software engineers are all together in the same page, in the same room, to understand where to optimally integrate the AI in the workflow.
Thank you very much, Dr. Tang. Do we have any more online questions?
Yes, we do. The next one is, what is the optimal approach for user feedback?
Oh, thanks. Yeah, so the user feedback I see, as I mentioned, I think it has to, it applies all across the AI development to deployment lifecycle. And so in the design and development stage, that's where the user feedback will be very helpful for defining the problem further, and also for iterative development of the AI tool. From a post-implementation standpoint, I think I alluded it to in parts of my presentation about the lifecycle maintenance aspect, where you would need to ensure that there's user feedback is captured, documented in a way that could help to either maintain or improve the AI system for PV over time.
Thank you. And Corey, I think we have time for one last question online, please. If we do.
All right, the next one is, what lessons have emerged so far on what makes human-in-the-loop oversight work well in practice? Any practical tips that you would share with teams that are designing it?
Yeah, I think so the question has to do with human in the loop and kind of best practices around how to effectively apply human in the loop in this with the AI systems. So in my experience, I think, and you'd have to think about really understanding from the end user's perspective, what are their pain points, and how you would how the AI would be used to solve that, and then how could it integrate in the workflow. And if you think about human in the loop as well, you have to think about it from a context of use and the model risk, right? If the model risk is high, which means that for that particular task, which means the decision consequence is high. As you know, in PV, we have a very low threshold for risk because we're concerned about missing a safety signal. We're concerned about not having timely reporting requirements. So if you insert the people where the human in the loop has the most impact, it would be at those places, like those critical points of where it could be a barrier to getting timely reporting, timely, accurate, and complete information so that you would meet those regulatory requirements. And really listen to the end users, because if a tool is useless unless they actually use it, and you can get a lot from understanding exactly where the human loop is, because essentially human in the loop is them figuring out how they're going to use the tool effectively, right? Thank you.
Right. Thank you very much, Dr. Dan. This brings us to the end of this day's events and to the end of this presentation. Thank you very much.
Thank you.
So we will officially adjourn for the day. We begin again tomorrow, same room at 9 a.m. Please remember that Dr. Dan and Commander Shields will be here for the one-to-ones. I know it's a long day, but if you do have questions, please avail yourself of this opportunity. And, they will be here as long as you need them to be here. Well, no longer than 4:50. So thank you all very much. Safe travels back to where you're going tonight, and we'll see you again tomorrow. Thank you.